Assalamualaikum, good morning and Salam Malaysia Madani. Before we begin, may I please request for the participants to please fill up the seats in front if possible. Thank you. Because there are still a lot of empty seats in front, so just help us fill up the front seats first. Assalamualaikum and Salam Malaysia Madani. Ladies and gentlemen, please take your seats as we prepare to begin our symposium, which will commence at 9 a.m. So before we begin, I would like to address a few housekeeping announcements. So first and foremost, we kindly request that all mobile phones and electronic devices be kept in silent mode throughout the symposium. Upon the announcement of the arrival of the VIPs, please rise and remain standing until the national anthem has concluded. All meals, tea breaks and refreshments will be served at the banquet hall on level 4. Please follow the signposts outside the auditorium for directions. In the event of an emergency, please note that emergency exits are located at the back of the auditorium and on either side of the stage. If necessary, please proceed to the nearest exit in a calm and orderly manner. So lastly, restrooms are located on the left side exit of the auditorium and the prayer room is located on level 3. Thank you for your kind cooperation and attention. announcing the arrival of Yang Berbahagia Datuk Dr. Nur Fariza binti Ngah, Deputy Director General of Health, Research and Technical Support, Ministry of Health, Malaysia. Please remain standing for our national anthem Negaraku. Please be seated. Yang Berbahagia Datuk Dr. Nur Fariza binti Ngah, Deputy Director General of Health, Research and Technical Support, Ministry of Health, Malaysia. Yang Berusaha Dr. Murizal bin Zainul, Manager, National Institutes of Health, NIH, Malaysia. Yang Berusaha Dr. Ami Fazlin binti Said Muhammad, Director, Institute for Medical Research, IMR, National Institutes of Health, Malaysia. Yang Berusaha Puan Safriah binti Mat Azhar, Registrar, National Institutes of Health, Malaysia. Yang Berusaha Dr. Toby Nagel, Founder and President, Phages for Global Health. Yang Berbahagia Datuk Dr. Suresh Kumar Chidambaram, Infectious Diseases Consultant, Sungai Buloh Hospital. Senior officers from the Ministry of Health, Ministry of Science, Technology and Innovation, and Ministry of Higher Education, Malaysia. Distinguished guests and speakers, honorable guests here with us today in the auditorium, as well as those joining online. Ladies and gentlemen, welcome to the Malaysian Stakeholders Symposium for Phage Therapy. I am Dr. Hadina from Institute for Clinical Research, NIH, and together with Dr. Sharifah Nurul Ain from CRC Hospital Sungai Buloh, we will be your MCs throughout this 1.5-day symposium. Before we begin, I would like to invite Yang Berusaha Dr. Hilmi bin Abdul Razak to recite the dua.
Assalamualaikum warahmatullahi wabarakatuh and good morning. Al-Fatiha. Bismillahirrahmanirrahim. Alhamdulillahi rabbil alamin wassalatu wassalamu ala asyrafil anbiya'i wal mursalin wa ala alihi wa sahbihi ajma'in. O Allah, praise be to God, Lord of the worlds. Peace and blessings be upon our Prophet Muhammad sallallahu alaihi wasallam and his companions, and for those who follow his example till the judgment day. O Allah, on this blessed morning, in conjunction with our program today, we beseech Thee and are grateful to You for all the infinite blessings bestowed upon us, Your humble servants, to live a safe and prosperous life. We seek Your blessing for flawless progress of this event from the beginning till the end. We seek Your guidance to steer clear of events that will be detrimental to the progress of this program. O Allah, bless us with Your tawfiq and hidayat. Please guide our institute to greatness, peace, glory, and prosperity. Guide us to become responsible intellectuals. Grant us valuable knowledge that will be beneficial to mankind. Make us your righteous servants who follow your commands and neglect sinful acts. Please forgive us for our wrongdoing. O Allah, bestow peace to our beloved country. Protect us from any threat and disaster, neither man-made nor natural. And to you, O Allah, we ask for security and prosperity upon us, our leaders, and our country. Bestow patience in us to face challenges from you. Please accept our deeds and please reward us accordingly. ربنا آتنا في الدنيا حسنة وفي الآخرة حسنة وقنا عذاب النار وصلى الله على سيدنا محمد وعلى آله وصحبه وبارك وسلم والحمد لله رب العالمين. Thank you.
Thank you, Yang Berusaha Dr. Hilmi. As we settle in, I trust that you have all received a physical or a digital copy of our agenda today. So as you can see, we have an incredible lineup of speakers, researchers, clinicians, industry leaders, and policymakers all coming together to explore and discuss the latest breakthroughs, challenges, and future directions in phage research, therapy, and regulation. This symposium is jointly organized by the Institute for Medical Research, IMR-NIH, together with Phages for Global Health, with collaborations from the Center of Excellence for Omics-Driven Computational Biodiversity, ComBio, AIMST University, as well as Institute for Clinical Research, ICR-NIH. Therefore, it is my great pleasure to invite Yang Berusaha Dr. Ami Fazlin binti Said Muhammad, Director of the Institute for Medical Research, IMR-NIH, as the main organizing body of today's symposium to deliver a welcome address.
Okey, yang berbahagia Datuk Dr. Nur Farizangah, Deputy Director General Research and Technical Support, Ministry of Health Malaysia. Yang berbahagia Datuk Dr. Suraj Kumar, anak lelaki Cidambaran, Infectious Disease Consultant, Sungai Buloh Hospital, Malaysia. Yang berusaha Dr. Muriza Zainul, Manager, National Institute of Health Malaysia. Yang berusaha Puan Safriah binti Muhammad Azhar, Registrar, National Institute of Health Malaysia. Associate Professor Dr. Sivachandran Parimanan, AIMST University. Dr. Toby Nagel, Founder and President of Phages. Senior Officers, Ministry of Health Malaysia, Ministry of Science, Technology and Innovation, and Ministry of Higher Education. Distinguished guests and delegates, ladies and gentlemen, Assalamualaikum warahmatullahi wabarakatuh, and a very good morning. It is with pleasure and enthusiasm that I welcome you to the symposium for the phage therapy here in the National Institute of Health, Alam. As we gather today, we are united by our shared passion and commitment to advancing the field of phage therapy, a promising frontier in the battle against antibiotic-resistant infections and the quest for innovative medical solutions. The significance of this symposium cannot be overstated. Phage therapy, the use of vector phages to treat bacterial infections, has seen a resurgence in the recent years, driven by the growing threat of antibiotic resistance. As conventional antibiotics becomes less effective, phage therapy offers a beacon of hope with its potential to provide targeted, effective, and personalized treatment for patients worldwide. This symposium also serves as a platform to celebrate the achievement and milestones in phage therapy, from pioneering research that has expanded our knowledge of phage-host interaction to clinical trials demonstrating the efficacy of phage treatment. However, there is still much work to be done. Over these 2 days, we will have the opportunity to delve into the latest research, share insights, and engage in meaningful discussions with experts from various disciplines. Our diverse lineup of speakers includes leading scientists, clinicians, and innovators who have made significant contribution to the field. Their work not only advances our understanding of phage biology, but also paves the way for practical application that can revolutionize healthcare. Ladies and gentlemen, esteemed colleagues and honored guests, as we embark on this intellectual journey, I encourage you to embrace the spirit of collaboration and curiosity. The challenges we face in combating antibiotic-resistant infections are multifaceted and complex. It is through the exchange of ideas, cross-disciplinary partnership, and collaborative efforts that we can overcome these hurdles and make meaningful progress. I would like to extend my heartfelt gratitude to the organizers, sponsors, as well as volunteers who have worked tirelessly to make this symposium a reality. Your dedication and hard work have ensured that we have a platform to come together, share our knowledge, and drive the field of phage therapy forward. To our speakers and presenters, thank you for sharing your expertise and insights with us. Your contributions are invaluable, and we are eager to learn from your experiences and findings. To the attendees, your presence here is a testament for your commitment to advance the phage therapy and improving patient outcome. I encourage you to take full advantage of the opportunities to do networking, engage in discussions, and explore new collaboration. As we embark on this symposium, let us remember that the upcoming work that we will do has the potential to change lives. By harnessing the power of bacteriophages, we can develop innovative treatments that offer hope to patients suffering from antibiotic-resistant infections. Together, we can make a difference and pave the way for a healthier future. Once again, welcome to the Symposium for Phage Therapy. I wish you all a productive, enlightening, and inspiring experience. Thank you.
Thank you, Yang Berusaha Dr. Ami Fazlin, for that wonderful welcome. The Symposium for Phage Therapy today brings together stakeholders from various corners of the industry, hoping to drive impactful research that would benefit patients, healthcare workers, and pharma industries alike. This has been made possible by resources and support from our own Ministry of Health. We are grateful to have with us today Yang Berbahagia Datuk Dr. Nur Fariza binti Ngah, the Deputy Director General of Health, Research and Technical Support, Ministry of Health Malaysia, as one of our key drivers of research and inter-industry collaborations. It is with great pleasure that I now invite Yang Berbahagia Datuk Dr. Nur Fariza binti Ngah to deliver the officiating speech.
Thank you very much, Madam MC. Yang Berusaha Dr. Murizal bin Zainal, Manager, National Institute of Health, Malaysia. Yang Berusaha Dr. Ami Fazlin binti Said Muhammad, Director, Institute of Medical Research, National Institute of Health, Malaysia. Yang Berusaha Pun Safriah binti Muhammad Azhar, Registrar for NIH. Yang Berusaha Dr. Toby Nagel, Founder and President of Phages for Global Health. Yang Berbahagia Dr. Suresh Kumar, Infectious Disease Consultant, Hospital Sungai Buloh, Senior Officers, Ministry of Health Malaysia, Senior Officers, Ministry of Science, Technology and Innovation Malaysia, Senior Officers of Ministry of Higher Education Malaysia, distinguished guests, speakers, ladies and gentlemen. Assalamualaikum warahmatullahi wabarakatuh, and very good morning to everyone. It is a great honor to welcome every one of you to this distinctive symposium on phage therapy, a topic that holds a significant alternative in the realm of modern medicine. As we gather here today, we hope that the revolutionized potential treatment against multi-resistant bacterial infections and to refine our approach in combating antimicrobial resistance. Phage therapy, the use of bacteriophages to target and destroy bacterial pathogens, is not a novel concept. It was discovered a very long time ago, even before discovery of antibiotics. However, it is only in recent years, with the rising threat of antibiotic-resistant bacteria, that phage therapy has gained renewed attention and interest from the scientific community. As we convene over the next 2 days, we will delve into the myriad facet of phage therapy, from the biological phage itself to the latest advancement in research and clinical application. We will hear from distinguished experts who will share their insights on the mechanism of phage action, establishment of phage therapy in individual countries, and the phage regulatory system are among topics to be delivered. In this symposium, our discussion will span 4 different scopes: the phage research and biobanking, the clinical application of phage, the phage regulatory framework, as well as the phage product and manufacturing. So we go from the basic to the most advanced of the phage therapy. So it is crucial to acknowledge the collaborative spirit that drives this field. When I was first briefed by Datuk Suresh about the phage therapy that we suppose or we need to have in Malaysia, it was a very excellent, even though it's short briefing, but something that I think Ministry of Health should take the challenge. We should do more than what we have doing now, so that we can tackle the issue of AMR, that actually Malaysia also looking into it. So it is, phage therapy is actually a multidisciplinary endeavor that brings together microbiologists, clinicians, pharmacologists, regulators, manufacturers, and policymakers. Policymakers is important to be involved at the early stage, so that whatever decision that we need to inform the ministry, we must have some evidence base. So ministry is looking forward for any challenge or any ideas in regards to a new policies, but of course we need the backup on the scientific evidence to show that it's going to be a benefit to the population. So it is through this collaboration that we can overcome the obstacles and lie ahead and pave the way for innovative solution to some of the most pressing healthcare challenges and antimicrobial resistance. So in the earlier discussion with Dr. Suresh, Dr. Toby as well, Dr. Ami, we know that we have to work together. We cannot come under one sector only. We have to be united. We have to be, all have the same ideas and challenge each other in regards to how we can improve the phage therapy, or how can we start phage therapy in Malaysia. So to go stronger, you have to be united. You cannot work alone. Of course, you can work alone, but it's going to be slow. So if we work together, we know that we can go far and even faster to make this happen. So let us also remember that patients whose life depend on the success of our effort. For many, especially those with chronic and drug-resistant infection, phage therapy represents a beacon of hope. Our work today is not just about advancing science, it is about making a tangible difference in the life of individuals and communities worldwide. And as we embark on this journey together, I encourage you to engage in discussion, share your knowledge and experience, and challenge conventional thinking. I know like Singapore and many other states already embarked into phage. So for Malaysia, we are just starting. So this is the opportunity that for our Malaysian colleagues to ask questions, to ask anything that you have in mind in regards to phage therapy, so that we can learn from the country that are already advanced in the phage therapy. So let this symposium be a catalyst for new ideas, partnership, and breakthrough that will propel this field of phage therapy forwards. In conclusion, I would like to express my deepest gratitude to all the organizers, the speakers, the participants, for all dedication and commitment to advancing phage therapy. Thank you for being here in this hall and even online. And your contribution are invaluable, and I'm confident that our collective efforts and our 2 days discussion will lead to a significant advancement in the fight against bacterial infection. So with that, thank you, and I wish all of you a productive and inspiring symposium. Assalamualaikum.
Thank you, Yang Berbahagia Datuk Dr. Nur Fariza. For your information, today's symposium is not only attended by experts physically here, but is also being streamed online for a wider audience reach. Ladies and gentlemen, without further ado, let's kick off our symposium today with the first speaker, Yang Berbahagia Datuk Dr. Suresh Kumar Chidambaram. Dr. Suresh Kumar is an infectious diseases consultant physician at Hospital Sungai Buloh, Malaysia, and is a leading expert in his area. He is renowned for his work in HIV and AIDS, emerging infections, and public health crisis management, including his contributions during the COVID-19 pandemic. With extensive experience in clinical practice, research, and teaching, he has played a pivotal role in advancing Malaysia's healthcare sector and has earned recognition both locally and internationally. Today, he will be speaking on setting the landscape regarding antimicrobial resistance, AMR, in Malaysia. Please join me in welcoming Yang Berbahagia Datuk Dr. Suresh Kumar to the stage.
Thanks. I don't recognize the person she's introducing, but I see a lot of researchers in the audience here, and my task is to convince you all that AMR is desperate enough that you all have to put in a lot of your effort into AMR research, number one. And then number two, a small side effect, you might be scared of getting admitted to hospitals, but that is not my intention, okay? Let me start. So many of us know, in infectious diseases, we know tuberculosis, in this part of the world, dengue, HIV as huge infectious diseases problem, right? And but a bit of a secret that many of us in the ID field and IPC field have kept from all of you is all those infections are very small when compared to hospital-acquired infections. So the way this is a graph from WHO classified it, tried to come up with a standard scale. The scale is DALY, disably adjusted life years. It means years of life lost plus years of life lost to disability. So that is the scale that they use to classify all the infections. As you can see here, hospital-acquired infections account for twice the burden of 32 other infectious diseases. But it's somehow lot less research and innovation goes into hospital-acquired infections compared to many other diseases in public health. So these are huge problems. A lot of people are getting disabled or died due to hospital-acquired infections. And in fact, so much so that, in UN General Assembly had a high-level meeting in 2024, in September 2024, just for AMR as this thing. And the call is to decrease the average life expectancy, loss of life expectancy by 1.8 years globally by 2035, meaning what they said is, so just like COVID, remember when COVID hit us, average life expectancy dropped. So they are saying now the AMR has reached a point where the life expectancy is going to drop by 1.8 years globally by 2035, and we need to do something for that. So we were not able to, for tuberculosis, for HIV, we were able to clearly tell you how many people died per year. But for hospital-acquired infections, we struggled to come up with a number. And in fact, research published in 2019 was the first time we could get a number for how many people died due to antimicrobial resistance as a number. And that was important for us to compare and contrast against other diseases. And so that forecasting model has been used now. And this is the data now which says how many people are going to die due to antimicrobial resistance moving forward, right? So these are attributable mortality due to resistance, and these are the numbers that they talk about, and majority are going to be in this, as you can see the graph on the majority, see the last one, 2050, the orange bar, majority of those people are going to be in the elderly. So many of us are going to be there at that particular juncture. If you're not convinced that we need to work on it, this is the graph that's going to tell you better work on it, because many of us are going to be in the orange graph when come 2050, right? And so a lot of the deaths are going to happen in that group, and that's the reason why there's going to be an increase in deaths. And generally, so I always thought, we are doing innovations and stuff like that, and so our sepsis rates will come down over the years. Again, the projections are saying the other way around. The amount of patients getting infection in this world is going to increase. Again, it is part of the reason because of aging, and as well as the immunosuppressions that we do moving forward. And so infections are going to increase, not decrease in the years to come. That is the projection, right? Okay. So what WHO did was, in this whole program, WHO classified bugs into critical groups, high-risk groups, and medium groups. That's how they classified. So critical groups are groups where these are important bugs, but we don't have good antibiotics. So that's what is a critical group, right? And so as you can see here, for hospital-acquired infections, there were two of them that comes out, carbapenem-resistant enterobacteriaceae, or CREs, commonly called CREs. The NDM was a popular one. And then Acinetobacter, carbapenem-resistant Acinetobacter. So these are two hospital-acquired ones, and also there is ESBL, Enterobacteriaceae is the other one, right? So looking at the two critical pathogens, and if you look at this Malaysian data, so there's a lab-based information system. We collect all the microbes from the hospitals, and it goes to a centralized database, and then it is categorized, and it is classified. The denominator that we use is the rates per 100 admissions, right? Rates per 100 admissions. As you can see here, I just there is the top blue line that you see. I'm not sure I can show that. Right? Okay. The blue line that you see, this is Acinetobacter, one of the critical pathogens. And then this, I have no idea what this color is, but okay. So this color is the one that is from ground up, it's just going up, is carbapenem-resistant Enterobacteriaceae. And so the two critical bugs that we are talking about are going the wrong direction for Malaysia and many part of the world, right? Okay. And so there's something about carbapenem resistance, right? Carbapenem is the drugs like meropenem, imipenem, and so these were considered, those days we used to call it gorilla penem. You know why it's gorilla penem? So those days we don't have to worry. A guy comes in he's very sick, you don't have to think. Just give him carbapenem, he'll get better. So it's like, you don't have to think. No need to think. So it's called gorilla penem, right? So it was an antibiotic which we use and patients just respond. So just throw it at the patient responds, we are all happy, right? So that's what carbapenem was. And suddenly now we are seeing carbapenem resistance. The antibiotic that was safe, easy to use, and usually works is now getting resistance. As you can see here, this is data compiled from IMR, again, blood in our national surveillance program, looking at blood isolates alone. Acinetobacter, the carbapenem resistance, 65%. But there are smaller number of isolates. You can see 9,000 isolates over the year. But E. coli and Klebsiella are 36,000 isolates that we see. And they are 5% to 1.2 to 7% resistance. So sort of carbapenem resistance is getting bigger and bigger and bigger, right? Depending on what you look at. And I'll tell you what's the significance of this carbapenem resistance. There's a study looking at bloodstream infections due to Enterobacteriaceae in low and middle income countries, the Panorama study. It's a multinational prospective cohort study. They looked at about 300 patients. They divided them into carbapenem sensitive and carbapenem resistant. Same bugs, similar bugs. One is carbapenem sensitive, one is carbapenem resistant. Look at immediately in-hospital mortality rates. In-hospital mortality rates, carbapenem sensitive 20%, carbapenem resistant 35%. So carbapenem, somehow when it became resistant, we have not found replacements for it, and mortality rates are shooting through the roof. 35% mortality in this particular study, and of course, increased length of stay, so the whole things are going to get worse, right? Another study that this time around, that there was a cohort study. There's a randomized control trials. You know when we do randomized control trials, we get the best patients together, and then we randomize them to study, monitor them very closely, make sure everything is fine. So there's a randomized control study comparing what we use these days for carbapenem resistant bugs, mainly polymyxins, antibiotic called polymyxin. Polymyxin plus a placebo, or polymyxin plus meropenem. So the study results is not important. What I want to show you is both the arms, mortality rate here, 43% one arm, 37% an arm. The study was a failure. The meropenem added no difference. But I want to show even in randomized controlled trials of carbapenem resistant pathogens, the mortality rates are extremely high. We're talking about 30 to 40% mortality rates. That's the kind of rates of infections that we are now, mortality rates that we are dealing with now, right? Okay. So what does it all mean to an individual patient that I see in my ward, right? So the 61-year-old farmer with diabetes, hypertension, chronic kidney disease, the standard metabolic profile that we get when patients get admitted. He's admitted with septic shock due to E. coli. And he gets to ICU. It takes us a month to fix him. It takes us a month to fix him. He gets fixed, and then he's sent to my ward, step-down ward, where I manage. And then he's got some pressure sores. We are dealing with it, and he's got a catheter, urinary catheter, because to protect the pressure sores. And he develops infection again. And the urine CNS this time grows this pseudomonas that is resistant to everything, right, that we have, and so I hesitate to treat this. I just remove the catheter, watch him, but he becomes more and more drowsy, and the CRP levels go up, and I have no chance but to treat him. I give him polymyxin. Within 3 days, he develops renal failure, and subsequently the family doesn't want dialysis and wants to go home, and so that he can pass on at home. And so this is what happens in a setting where we have got drug-resistant bugs, and we don't have safe drugs anymore, and we've got drugs with lots of side effects that we are now forced to use to treat patients, and the adverse event happens, right? And of course, it's always drug innovation, right? The new drugs are always the answer for this, and true enough, the scientific community has met the challenge, and the series of new drugs have come in to treat these difficult to treat infections, right? But however, these are the bugs that we have mostly in Malaysia, or this part of the world. These are in carbapenem resistant enterobacterus. We have CREs and we have Acinetobacters. But many of these new drugs don't work for these conditions. And so while there is new drugs available, but they are not specifically to the needs of the country moving forward, and many of these don't work like that. Okay? And the second big problem is, of course, newer drugs can come in. Or newer drugs are coming in, but access to newer drugs is always difficult in a small country like Malaysia. So I'm giving an example. Ceftazidime avibactam, FDA approved in 2015, 2016. We have it now, but all other bugs you can see, they were FDA approved or European Medical Authority approved many years ago, but they are still not available in Malaysia. People don't think it is worth to bring it into a small country like Malaysia. And in fact, even if they come in, the drug companies have learned something, the antibiotic manufacturers have learned something from oncology units, and the antibiotic prices are now similar to oncology drug prices. And for example, ceftazidime avibactam will set us down by about ringgit 4,000 per week, and that's the kind of strategy, low volume, high cost strategy is what they're using it for new antibiotics. And so new antibiotics got a problem. In fact, we are working with this group called GARPI, and GARPI has come up with a projection saying that, because of carbapenem resistant infections, right? While there will be 1.59 million people with carbapenem resistant infections, because of access barriers, almost 95% of them won't get access to good antibiotics, and then only 85,000 of them only 85,000 of them will get appropriate antibiotics, and 95% won't get appropriate antibiotics because of access problems. So having antibiotics is fine, access is also a big issue. And then just to zoom in on one particular bug to highlight what's happening in the treatment field in hospitals, I will just focus on Acinetobacter, right? Just tell you what treatment options we have, and I'll show you why the situation is bad. Okay, so this is our national consensus guidelines for treatment of multi-drug resistance organisms that we published in 2024, and we say if you have moderate to severe infection due to Acinetobacter, meaning if you have a pneumonia, if you've got a bloodstream infections, you have to use ampicillin sulbactam, high dose, there is 9 grams of sulbactam per day. FDA approved dose is 6 grams, but we are using very high doses for which we, not many of us have got enough data for it, but that's what we're going to use. We are pulling out an old drug called minocycline, which we steal it from dermatologists. Dermatologists use it for acne, and suddenly we take it from them. We have only oral, we don't have the IV because we need to search for it. And then we use a drug that I showed you earlier, polymyxin, which can cause kidney problems, right? So that's a combination that we are supposed to use, right? And if somebody has got bloodstream infection or urinary tract infection, we can't use minocycline because it has, where is my pointer? Okay, fine. Minocycline has got low serum and urinary levels, right? And so we are restricted on what we can use, right? Okay. So polymyxin becomes very important drug here because ampicillin sulbactam is resistant. So ampicillin sulbactam in a lab is resistant. But we are using super high doses to try and overcome the resistance, but we use it in combination, right? So we're using in combination. So we're using a drug that's already resistant, but we're using super high doses. Then we have another drug, polymyxin B, right? So polymyxin B or colistin, whichever it's, now if the lab does breakpoints or they do susceptibility testing, the lab will tell us it is intermediate or resistant. There will be no lab report says it is sensitive. So we're going to use a drug that is now intermediate at best. So it's not sensitive, right? And you can see here, why is that so? So CLSI, that determines breakpoints for us, says we won't give any more breakpoints for polymyxin. Sensitive breakpoints for polymyxin. We only give intermediate breakpoint for polymyxin. It means you have to use polymyxin or colistin in this with another active antimicrobial. The problem is we don't have another active antimicrobial. That's why using super high doses of ampicillin sulbactam that is already resistant. Okay? And then they say, even when you do it, it is unlikely to be effective for pneumonia. So CLSI says this. But every day I see a pneumonia patient with Acinetobacter in my ICU, I'm using this. This is what I'm using it. I have nothing else. Right? And of course, way back in 2015, there was a call for urgent action of polymyxin Acinetobacter baumannii, which we don't have any treatment currently. And when we did a short study looking at all of our bloodstream infections during just after COVID, we found about 50% of bloodstream infection of Acinetobacter is actually resistant to polymyxin. And so we do already have this, and just because polymyxin susceptibility testing is not routinely done, we thankfully don't know how much of polymyxin resistant we have currently. Okay, so to highlight this problem, that's what this is a paper from our colleagues from University of Malaya that looked at carbapenem-resistant Acinetobacter bacteria, and they looked at in the group that they saw about 90 patients, 60% of them were infections, 40% were colonizers. The 60% that were infection, their mortality rate was 57.4%. But it's always difficult to understand mortality for hospital-acquired infections because even 40% of the Acinetobacter is a colonizer. It means a colonizer is 30% infection, 30% resistance, 30% mortality, sorry. So these infections happen in people who already have high mortality rates, right? And then on top of it, when this bug comes up, the mortality rate increases. And so that's why you see such huge numbers of mortality rates coming up, right? Okay. Yes, new drugs are coming in for Acinetobacter, for example. There is a drug called sulbactam durlobactam that has now been approved in US FDA for pneumonias and hospital-acquired infections. Just came out in 2023. And as you can see, even the trial, the standard of care, you can see for colistin is 32% mortality rate for colistin. And so even in clinical trials, again, I'm showing you 1/3 of these patients die when we use the current standard of care in Malaysia, that is 32%, right? But of course, as I've showed you earlier, new antibiotics can come, but when will it reach Malaysia is another different story. And how much it's going to cost us is another different story. And so to summarize, AMR landscape it's a silent pandemic because many people outside the circle may not know how much it kills. And I think that's something that we need to do a better job. We shouldn't. I was told by expert, climate change, it took 10 to 15 years before scientists knew about it and people started worrying about it. So AMR, we need to do it faster. We really have to do it faster. And despite all the technological advancements we are doing, it is going to get worse. For example, because of aging and stuff like that. Access to effective and safe antibiotic is challenging because of the difficult bugs as well as access issues. We will be seeing some gains due to better infection control. This is pushing us to do better infection control, but still we need new approaches. With that, I'll thank you all.
Thank you, Yang Berbahagia Datuk Dr. Suresh Kumar, for that insightful talk and for appropriately scaring us of the dangers of AMR. But some might say that fear is the greatest motivator, and I hope it motivates all of us to work even harder to overcome this challenge. Our next speaker is Yang Berusaha Associate Professor Dr. Siva Chandran Parimanan from the Centre of Excellence for Omics-Driven Computational Biodiversity, COMBIO, at AIMST University. Associate Professor Dr. Siva Chandran is a distinguished researcher specializing in bioinformatics, AMR, surveillance, phage genomics, and bacteriophage therapeutics. As the head of the Research Management Center at AIMST University, he leads research and development initiatives, manages research grants, and fosters collaborations with both local and international institutions. Additionally, he plays an active role as an instructor in the Phages for Global Health workshop series, contributing to capacity-building efforts in bacteriophage research worldwide. Please welcome Yang Berusaha Associate Professor Dr. Siva Chandran Parimanan to give us an introduction to phage biology and an overview of phage research in Malaysia.
Thank you so much. I would like to, a very good morning to all the distinguished guests and all the participants of this symposium. So so I'm tasked to actually give you an introduction about phage biology and overview of phage research in Malaysia. So just a quick background about myself. My name is, I know the person has introduced, but I just want to mention about our institute. My name is Siva Chandran. I'm from AIMST University. I'm part of the Center for Excellence for Omics-Driven Computational Biodiscovery. So under that, we have an arm called Phage. So we have been working a lot on phage research, actively collaborating with a lot of phage researchers around the world, and that's how we got in touch with Dr. Toby, and today we are here, gathered to, I was actually shocked that it happened so fast. So thank you all for coming. So, and I hope I do justice. Bear with me if I'm going too basic. I'm not sure, because many of you come from different parts of the spectrum, right? So some may know more, some may know less. So I may go very basic, so bear with me. Thank you. All right, so I'm required to show this every time I make a presentation. This is our campus. This is an aerial view of our campus. We are in Kedah, in a town called Bidong. All right, so I think this has been oversubscribed every time we speak about this. The bottom line is AMR infection is here to stay. We need to do something about it so that we can bring down the mortality rate. Because it's predicted, WHO has predicted by 2050 to be one of the leading cause of death, amounting about 10 million deaths annually. So we need to do something about it. And bacteria adapt so fast, right? So we need to do something about it. All right. So what are phages, right? They are basically viruses. And after COVID, whenever I say virus, there's a panic look in the face, right? So don't worry about this virus. This virus does not harm humans. It only kills bacteria because like how COVID, humans, animals are the hosts. For bacteriophages, the bacteria is the host for it. And it naturally exists in our body. It's not something that we dig and bring out. It is already naturally existing in our bodies, food, in the environment. It does not harm human cells. It can kill both antibiotic resistant and sensitive bacteria. So it doesn't discriminate. It doesn't selectively only target the antibiotic resistant bacteria. It can also kill the sensitive ones. But it only kills specific bacteria. When I say specific, narrow spectrum, because phages will only kill the bacteria you're targeting, right? It will not kill like antibiotics, you have broad spectrum. So this is a more targeted spectrum. And you can actually design it to minimize future bacterial resistance. I'll talk more about this, and definitely later I think the other speakers will speak more about this as well. And the difference between an antibiotic and this, it can self-replicate. So antibiotic is a drug, right? It has its half-life, you have to do repeated dosing and all that. But bacteriophages are viruses which can self-replicate. As long as the host is there, it can self-replicate. But I'm not saying that does not mean that there doesn't need to be any repeated dosing. It may not need to have that much repeated dosing as an antibiotic, or maybe may not require repeated dosing as well. So this is something, a lot of things needs to be looked at during phage therapy. All right, so I try to make a very simple animation to put things in perspective. So when you use antibiotics, right, so whether it's bad bacteria or good bacteria, it kills everything. It does not discriminate, right? Whereas phages, you actually kind of source phages that are specific to target the bad bacteria, the bacteria that you want to eliminate. So it only targets those bacteria and only kills them. So it doesn't harm the natural commensals in your body. So that's why I would like to say that not all viruses are bad, right? Some viruses can be our friend and our ally to combat antimicrobial resistance, right? So where do we get these phages from? So these phages are everywhere. They're one of the most abundant most abundant organism. We can source them from environmental samples like sewage water, soil or water sample. And this water sample could be potentially where you suspect these bacteria are inhabiting, the bacteria you want to target, or maybe it could be areas where it is contaminated by this bacteria that you want to target. And let me say sewage water is an excellent source to get, bacteriophages. I will show you some data which will support this as well. So now the next question, are phages safe? In general, yes, and that is why FDA has actually given it generally recognized as safe status, where it is permitted to be used on ready-to-eat food. So it is generally safe. Okay, so history of phages. I think just now Dr. Nur Fariza Binti Nga has already told us it is not something new. It has been existing since 1919. So it was used as antimicrobial agent before antibiotics existed. Right? And then it was used in US, Europe, Asia, and very actively used in the former Soviet Union. So for more than 100 years it has been used. And in certain countries like Georgia, it is still being actively used as an antimicrobial agent. And now, because of the AMR crisis, it's being revisited in US, Europe, Australia, Middle East, and it is used in many different areas, not just therapy in the healthcare sector. It's used for food decontamination. There are more than 2,000 emergency clinical cases under compassionate use that has used bacteriophages to treat the patients. At least 35 clinical trials have been initiated from 16 countries, and there's no major adverse event that has been reported. So, as I was mentioning, in Georgia, more than 100 years, this is the center that has actually been there treating people with phages for more than 100 years, called the Eliava Phage Therapy Center. It's effective to treat. It has been used in many medical specialties, and it can treat antibiotic-resistant infection. So now, moving to the basic. So this is like a representation of a bacteriophage structure. But there are many different types of bacteriophage structure. This is just one example of it. This is a T4-like phage, and it has that capsid, the head, isocohedral head, and then you have the collar, the sheath, and then base plate, and the tail fibers. So the tail fibers are important because that's the one which recognizes the receptor on the surface of the bacteria, and then after that it starts the process of invading the cell and killing it. So the life cycle that phages undergo, there's two, is lytic and lysogenic. I will visit lysogenic after this, but now I will talk about lytic. So basically what happens is, like I mentioned just now, the phage, the tail fibers of the phage will kind of land on the bacteria. It get adsorbed to the bacterial surface, and then it will inject the DNA into the host cell, and then it takes over the host cell machinery and starts producing viral components, right? So then that viral components gets assembled, you get mature phages, and then you have like enzymes like holins and lysins being produced. The holins basically kind of makes holes in the inner membrane of the bacteria, and then you have gives access to the lysins to actually go and destabilize the peptidoglycan layer, and then because now the structure is destabilized, the phages through osmotic pressure can actually, burst open and come out of the bacteria and these resulting ones are known as progeny phages. Now the cycle repeats. The progeny phages continue to, in fact, that's why you say self-replicating. All right, so this is a transmission electron microscopy image of how the phages are attaching to the surface of the bacteria. And then I was mentioning about the lytic cycle. They also have another cycle called the lysogenic cycle. So not if they are lytic phages, they don't really go through the lysogenic cycle, but there are certain phages known as temperate phages or lysogenic phages. They have certain properties, certain genes that what happens is when they invade the cell, the DNA does not take over the host cell machinery. Instead, it goes and incorporates into the host genome, forming a prophage. So what happens is it just remains dormant there until some selective pressure, some induction happens. Then it may undergo lytic cycle, but these lysogenic phages are the least desirable ones for phage therapy. The lytic ones are the ones which are preferred. So this is why, later I'll be mentioning, having genomic sequences of the phages is important for us to analyze, to know whether these phages have any undesirable genes in them. So this is why we actually do phage genomics. All right, so later I think Dr. Jeremy will be talking about what do you call that, resensitization of resensitization of bacteria to antibiotics. So I'll be giving a little, just an introduction, and then later Dr. Jeremy will explain more about it. So now, when we are going to use phages, there are a few things you need to consider. One of it is, if you're going to use a single phage, bacteria can still delete the receptor which the phage identifies, and that phage can be resistant to the bacteria. So what I'm trying to say here is that just because we have phage doesn't mean there's not going to be, there's not going to be any resistance against the phage. Phage is like the ultimate, nothing can be done, the bacteria cannot fight the phage, it can. So there are also things that you need to do to ensure that you don't have cross-resistance of the bacteria to the phages. So in this case, most of the times phages are administered in a cocktail where you have mixed phages, and each of these phage will target a different receptor on the bacteria. So when the bacteria mutates itself, still you have the other phages that are still able to kill the bacteria. Right? So I think a lot of them later will be talking to you, and they'll be mentioning a lot of them actually use phage cocktails, part of the treatment. All right, so this is what, I think this is actually from Dr. Jeremy's, this is not from you, oh sorry, I think the later one, I think. Yeah, so these are some work that has been done where they showed after exposing the bacteria to phages, there has been resensitization of that bacteria to antibiotic once again. So basically, like, there's many resistant mechanisms. So one of it is the antibiotic efflux pump. So basically what it does, it just flushes out the antibiotics. So what happens is when this particular bacteria was treated with phage, it actually deleted its efflux pump. Of course, it became resistant to that phage, but it became sensitive to that antibiotic. So this was observed in Pseudomonas aeruginosa. Similar thing was also observed in A. baumannii, but not the efflux pump. Instead, this was a polysaccharide capsule. So this basically protects the bacteria from antibiotics. So when they treated with a particular phage, it actually deleted the capsule gene, and then it became phage resistant, but more sensitive to antibiotics. So this is an image from that paper. It showed that the wild-type bacteria, before exposing the phage, it has that polysaccharide complex, but then after it became phage resistant, that complex is missing. The capsule is missing, sorry. So now, I've given you an introduction about phages and all the, what are the different approaches, how they source the phages, what are the things you need to consider. Now, I would like to talk a bit on genomic surveillance, the importance of it. So genomic surveillance, here there are two things we are talking about. One, we are talking about first doing a surveillance on the AMR bacteria itself, for us to understand the resistome landscape, to understand the whole scenario, the evolution, the process. So this will not only just benefit phage therapy, it also benefits in better decision making towards enhancing antibiotic stewardship. So you can track resistant gene, understand the resistance evolution, and you can actually use this information to have a more targeted antibiotic therapy and plan strategies to control infections in hospital. But it's also very useful in identifying suitable targets. By knowing the bacterial genome, you can also use that information to identify suitable phage targets. But that is for the bacterial sequencing. You also need to sequence the phages, because you need to know, one is you need to understand whether the phage has any undesirable genes. It could be carrying antibiotic-resistant genes. The phages can carry antibiotic-resistant genes. It can be carrying virulence factors. It could be having, lysogeny-related genes, like integrases and all that. So we need to be sure, okay, this phage the genome is clear, we can use it. And at the same time, we also need to, when we make a cocktail of phages, we need to select phages with diverse genomes. Having two phages with the same genome means it has the same receptor. So if the bacteria deletes the receptor, all the phages in the cocktail become ineffective, right? So you need to make sure it's of diverse genomes so that, if it mutates against one, you still have other phages in the cocktail that can still kill the bacteria. So these are the things that needs to be looked into. All right. So after this, I'm done introducing the concepts. Now I want to move towards some of the data that we have. So we were part of this big initiative called the Global Pathogen Surveillance Project, together with WHO, Novo Nordisk, Denmark. It was done by Technical University of Denmark. So this started in the year 2016. So basically, it was a proof of concept where even before, when COVID happened, everyone was jumping the bandwagon to take sewage sample, sequence it to see whether COVID is there or not. So this was prior to that. So this started in 2016, where it was a proof of concept for applying metagenomics approach to initiate a global surveillance of human infectious diseases, including antimicrobial resistance from sewage. Because when you collect data from hospital, you're only having hospital data, but you want population level data. Sewage is a very excellent source to get population level data. So we collected, in Malaysia, we collected from major cities like Kuala Lumpur, Penang. I think we also had from Kedah, a few locations in KL and stuff like that. So, this is not very important, but the next point is very important. What we found is that the AMR levels in South Asia were higher than in Europe. That was one important finding. Another one thing that we saw is that there was none of the dominant AMR genes which are known to be restricted to a particular bacterial genera. That means any bacterial genera can be carrying the resistant gene because due to horizontal gene transfer. So which means things that may not be a pathogen today may be a pathogen tomorrow. So these are worrying trends that we observe. And the other thing is we also, because it's from metagenomic sequencing, so we can get viruses, we can get bacteria, so we got everything. And we actually analyzed the virome fraction. And the sewage water, the largest fraction of viruses were bacteriophages. So it's basically a powerhouse full of bacteriophages. So the source is there, it's just sourcing it, finding the right host, and going forward. All right, so these are some of the, so I was actually shocked. I never knew there were so many phage researchers. So that was something I liked when Dr. Nur Fariza was mentioning just now. We need to come together. So there were so many of us, we just didn't know each other. So I hope this platform will be a good platform for us to start collaborating. Instead of competing, we complement each other, so more gets done with the funds that we get. These were all grant-funded projects, actually. So they were from various universities, private and public universities, and they were targeting against E. coli, Pseudomonas species, Salmonella enterica, Staphylococcus, Xanthomonas. So it was varied. It was not only for human health. Human health, it was not for phage therapy yet. It was more of isolating phages, building the library. So more towards phage-host interactions and more of isolation and characterization of phages. And a lot of work was being done for aquaculture, using phages as an alternative to antibiotics in aquaculture, food safety, agriculture, and also animal health. So a little bit on the focus at our center right now. So what we are doing is we are trying to be, mimic the One Health approach. So we are trying to try to build a collection of well-characterized phages against animal, plant, human pathogens. Human pathogens primarily focus on escape pathogens as an alternative to antibiotics. So a very quick overview. So those phages for healthcare purposes, we are only at the level of isolating and just keeping them to be part of our biobank. But where we have a lot more progress is in the area of agriculture. We have actually developed a product where you can use phages as an alternative antimicrobial against plant pathogens. We have done in aquaculture as well, and also poultry, we have against salmonella. So now we are doing the efficacy and safety of phage cocktails in salmonella infected animal model and human cell lines. So hopefully we are hoping to see whether we can apply this in the poultry industry. And of course, a large part of our work is we are in collaboration with University of Copenhagen and University of Leicester. We are part of this initiative called the Phage Compass, where we actually develop tools for phage genomics, develop bioinformatics analysis tools for, so researchers don't have to do a lot. They can just submit the sequence there and then you get data on taxonomy, on whether or not it's suitable for therapy. So we are building tools in that. And of course, we work a lot on bacterial genome surveillance. We actually have started several projects with CRCs all the CRCs to look at Abamani surveillance, genome surveillance, and all that. So my take, I feel that phages should not be just looked at for therapy in the healthcare sector, but we should be also looking at it as in a One Health approach, where other places which uses antibiotics, whether can we reduce the reliance on antibiotics in those areas, like whether it's the poultry or whether it's the livestock farming and other areas as well. So this is something that we strongly believe. So like we can use it in ready-to-eat food to enhance food safety, animal feed to enhance immunity, and crops for protecting them and prolonging shelf life. So that's it from me. Thank you. Sorry if I was a bit slow, but I was just trying to pace myself, trying to explain. So I also have to acknowledge my team. Of course, I'm the face presenting, but these are the people who actually made it happen. And also, I have to thank a lot of the funding bodies which actually funded a lot of our work. And that's it. Thank you. So that's all.
Thank you, Yang Berusaha Associate Professor Dr. Siva Chandram, for that enlightening crash course on phages and a rather optimistic insight into tackling the AMR crisis. So our last speaker before we break for the morning is Yang Berusaha Dr. Vignesh Sivanathan. Dr. Vignesh is a biochemist and microbiologist who leads the Science Education Alliance Phages program at the Howard Hughes Medical Institute, HHMI, in Maryland, United States. The SEA program is known for their robust integration of authentic research experiences into undergraduate teaching, enabling undergraduate students to engage in high-impact and inclusive research projects. Through the Science Education Alliance, students at HHMI are able to participate in groundbreaking research, uncovering novel insights into phage biology, defense systems, and therapeutic applications. To give us a more in-depth insight into the C Phages program, please welcome Yang Berusaha Dr. Vignesh Sivanathan.
Sorry. Okay, we'll start again. Salam Sejahtera dan selamat pagi. Good morning, everyone. I'm Vignesh Sivanathan. Berasal dari Klang, but right now I'm at the Howard Hughes Medical Institute in the United States. HHMI, if you're not familiar with it, is a private biomedical research institute and funding agency in the US, and it's actually the largest private biomedical research institute and funding agency. HHMI is committed to advancing both science, but also science training, so science education in the US. And the program that I lead is called the Science Education Alliance. Dr. Toby invited me to speak about the Science Education Alliance because it offers you an opportunity to think about, as you go to develop a phage bank, how could you do that in a way that builds capacity in Malaysia for phage research in the future? So you can think about building a phage bank now, but where do you want to be in 10 years from now? And so I'll tell you about the work that we do in the Science Education Alliance, and then you can think about how you might apply that in Malaysia. Okay, so I'm going to walk through the goals of the program, the approach that we've taken, and then we'll talk about some of the outcomes of our approach. And so in the US, this program, the Science Education Alliance, was started really to enhance the scientific training and research capacity at universities in the United States. And so we think about who does research, right? So we think research is typically done at universities, and if you go to a university and look at who are the ones there are 4 groups, 4 constituents that make up the primary bodies at universities. Those are undergraduates, your postgraduates, your postdocs, and your STEM faculty. Of these 4 groups, the ones that are actually doing research at universities are the postdocs, the postgrads, and the faculty. The undergraduates are not really involved in research. They're just there for training, right? And so when we look at this, it looks fine, right? It's a big proportion of the folks at universities that are involved in research. But when we look at the numbers of these various groups, we realize that only a small fraction of those at universities are actually doing the research. The largest body of people at universities, undergraduates, are not involved in training, right? They are the most junior science trainees. They are being mentored by scientists, but they're not actually doing research. And we understand why that's the case, right? And so, typically when we think of research, you're thinking about a research lab. You have a PI, a STEM faculty member, and you have a couple of postdocs, you have a couple of graduate students. And that's how research is done. Undergraduates make up a very large proportion, and there simply isn't enough STEM faculty, and there simply isn't enough space in research labs to actually involve or include these undergraduate students in research. And so as a result, we've devised an education system, a science training system that it's not like what we do in universities, not what we do as scientists, but one that looks more like what we do in schools, right? So we have lectures where we talk to students about science, and then we have teaching labs where we show them how to do science, how to do research, but they're not actually doing research. We teach them the skills, right? How to pipette, how to look at the results, how to interpret results, but they're not doing research. And so the question is, can we enhance science training and research capacity by integrating research into the undergraduate curriculum, right? So the undergraduate teaching labs have enough space and are designed to accommodate all these undergraduate students. If we can take the research from a funded lab, a research lab, and put it in the undergraduate teaching lab, then now we might be able to tap into the undergraduate students as part of the research ecosystem, right? So this not only trains undergraduate students to do research, but adds to the research capacity of universities or the US in this case. Okay, so how do you make that happen? And so the approach that we've taken leverages what we call massively parallel science. And so even if you go into a teaching lab, you might have 20 students, you can't actually have them do 20 different projects like you would your postdocs and graduate students in your lab. You simply can't mentor 20 different projects, especially by very novice scientists. But if you design a massively parallel project, so we know this term massively parallel from sequencing, right? You throw in lots of different DNA sequences, molecules, and you have all of them sequenced simultaneously in one process. And so if you can design a project that can be implemented in parallel by many students, but all doing their own independent work, now you can tap into massively parallel science. And so in thinking about projects that work well for this, phage discovery is one of them. And so I'll talk to you about how we've set this up. And so across the US, a teaching faculty or STEM faculty in their undergraduate teaching lab will have their students go out and collect an environmental sample. We don't send them to get sewage because we want it to be safe, and so these are undergraduates in teaching labs. But you can basically, as Siva mentioned, bacteriophages are everywhere on your skin and the environment. And so they go and they collect their own samples. They extract these, they try to extract any viruses that are in this sample, and then they look to see if any of them have phages specific to bacteria that we're interested in. And so here too, you're not going to give them a pathogenic bacteria. You don't want your students to be working with it, but you can use a closely related non-pathogenic version of the bacteria that you're interested in. So in the US, we've partnered with Graham Hatfull, who's interested in studying mycobacteria. And so we use Mycobacteria smegmatis as the bacteria that we give our students, non-pathogenic, but related to those that cause tuberculosis and other mycobacterial infections. Okay, so students plate these viral samples, they look for things that can infect, so they found now some phages. They go through multiple rounds of purifying it, so they have one phage per student, and then that sample, we start characterizing it. So they do some electron microscopy, but most importantly, as Siva mentioned, they also extract that DNA and we have it sequenced for them. Again, you can leverage now massively parallel students finding phages to massively parallel sequencing, so we can get tons of these phages that are sequenced. And then they go ahead and then they carefully annotate those genomes, right? Now, what's great here is that you don't just have a collection of phages, but for every phage a student finds, they are sending it in to us, and we are collecting it in our own phage bank. And so for every phage, we have the viral copy that we can go back to, and also the sequence that we can go back to. Okay, so in the US, we didn't want to limit ourselves to folks that were just phage biologists, and so we developed a mechanism or a process to allow any faculty member, someone who teaches basic biology, to be able to do this research with their students. Often it's these faculty who are teaching undergraduate students. And so we host, once a year, we host a training workshop. It's just a week long. It's, I think, very similar to what many folks here have done in the past. You train these faculty to basically do the protocols that the students are going to be doing. We standardize the protocols so that everyone, doesn't matter where they're doing it, is doing the same thing, and so that when we get the data back, we know how it was prepared. And then every year we also host an annual meeting where everybody comes back. We get to discuss, everyone comes back, we talk about our findings, we understand what needs to be done, we go back and we change what we're doing based on our discoveries. And what I want to show you is that this approach works. Right? And so when we started this program in 2008, we invited about professors from about 10 universities in the US. We trained them, they went back, and each one of them had about 20 to 30 students. So now in 2008, we have about 300 students going out to find bacteriophages for us. The next year, we just train a new set of schools, right? So we bring in another 15 to 20 schools. We train those faculty go back, and now in 2009, we have about 30 faculty who are now working with about over 500 students going and finding these viruses, sending it back to us. And this continues every year. That right now, so this is a little bit out of date, but in 2025, we have about 200 universities, 400 faculty, leading about 7,000 students to find these viruses for us every year, right? And that number is just going to keep going up. You see that little dip, that was COVID, and I think all graphs have that little dip. Okay, and so what are the outcomes of this approach? And so for one, I think the one that's most relevant here is that I think we have the largest phage bank in the world. Is that right, Toby? Yeah, I think the biggest ones are usually in the thousands, but here we have 28,000. Importantly, these were all isolated by undergraduate students who now have an understanding of research and microbiology and phage biology, and are also enthusiastic about it. It has allowed, this database is publicly available, so it's called PhagesDB. People request it, but we also partner with researchers in the US. And so this has now fueled a lot of basic research into phage biology. So we have really deep understanding of phage population structures, phage defense systems, phage evolution. And then again, more importantly here, I guess, is that these phages are also now routinely being, the phage bank are routinely being screened for phages that can be used in these compassionate cases for antibiotic-resistant infections. And so here are just some of the more recent papers describing some of these effects. Okay, now, so if you're thinking about building a phage bank in Malaysia, I'm hoping you'll see that this is a pretty good way of building a very big phage bank very quickly for your purposes. But in addition, I think where it becomes really valuable is that when we start projecting into the future, where do you want to be in 5 and 10 years? And so when we started the program, we didn't quite appreciate that now we have a community of over 400 STEM faculty who all have training in phage biology, who are all interested in continuing to do this work in phage biology. And in fact, it's because of them, right? So this is the research project that I just talked about. We call it C-Phages, not Southeast Asia, which I wish it was true, but it stands for the Science Education Alliance, and it's our phages project. So that's the discovery, we sequence, and we do the bioinformatic analysis. But because we have this large group of faculty who are interested in phage biology, they've pushed us, they want to do more. So we've developed an extra course that they're now doing with their students that we call, that created PhagesDB. This is called C-Genes, where for each phage, students are now cloning every gene in the phage genome and creating an expression vector so that we can take it, put it back into the host, and look for the effects of overexpressing any particular phage gene in the host. We're looking for phage genes that are going to shut down host metabolism and kill the bacterium. And this project has been running for just 5 years, and now we have a whole database of potential antibiotic targets. Sorry, I missed a part. So in addition to looking for phage genes that can stop bacterial growth, they then also do an interaction assay to look at which host protein the phage protein is interacting with. And so identifying a target that we can even use to develop antibiotics. And so now we have a second database called GenesDB that is also publicly available in the US, and people are requesting for it. After 5 years, those STEM faculty are like, okay, we've got this, what's next? And so right now we are in the process of developing yet another project which we call DeepC, which is basically going to use AI and all the data generated from both the phages and genes database to start modeling infection. So we're going to try to use AI to model those phage host interactions, both at the sort of organismal level, right? So specificity, but also at the protein level, looking for antibacterial targets. Okay, and so this new database will also become publicly available. Now, this started off as a project to engage students in training and also begin to leverage them to support our research capacity. And in 10 years, this program now has sort of national attention in the US, and they're trying to replicate this model. And in fact, at HHMI, we're also basically going to try to take over undergraduate education and make it research-based, because the students also learn so much better and are much more prepared for graduate work when you engage them in research early on. And so all that to say, as you think about developing your phage bank in Malaysia, I hope you'll consider integrating this into the undergraduate curriculum, both to enhance their training, but also to build capacity for phage research here in Malaysia, in the future. And so I'm going to stop. I'll take any questions if there are any. Yes. Yeah, no, and I think that's a really key thing, right? We see how it is so easy for foreign governments to pull back aid, right, from the rest of the world, and so developing this for ourselves, I think, would be critical. And this program is not being cut. So we are a private institution, and in fact, the government funding for this program, the government is actually partnering with our program to expand it as well. Really, because in the US, we also have a problem of science, persistence in science. Students come to the university thinking they're going to learn how to do science, but then they find themselves sitting in a lecture more like high school. And so 60% of students who go to universities in the US wanting to get a degree in the sciences will leave the science. That's a huge loss for the scientific workforce in the US. So this program has shown that by offering this type of training, there's real mentorship, that there's a significant boost even in their persistence in the sciences. Okay, thank you.
Thank you, Yang Berusaha Head Dr. Vignesh, for sharing with us that revolutionary approach pioneered by HHMI. Ladies and gentlemen, before we continue with our symposium, we will be taking a quick morning break for some refreshments. I would hereby like to invite all of you to proceed to the banquet hall on level 4 to enjoy some light snacks and drinks, and we shall resume the morning session at 10:50 a.m. Thank you and enjoy.
Welcome back, ladies and gentlemen. It is my great pleasure to introduce our next speaker, who will present on the overview of Australia's approach to establishing a national phage therapy system, financial model for economic benefit to Australia, clinical case studies. Professor Jonathan Iredell is a renowned infectious disease physician and clinical microbiologist with expertise in critical infections and antibiotic resistance. He has led the NIH Center of Research Excellence in Critical Infectious Diseases and has significantly contributed to antimicrobial stewardship, clinical education, and national policies on antibiotic use. Prof. John also spearheads gram-negative surveillance outcomes for the Australian Group on Antimicrobial Resistance and has served on several key advisory committees. Please join me in welcoming Professor Jonathan.
Thank you. Thank you very much for the introduction and for the invitation. I'm very grateful to our esteemed hosts for inviting me to speak. It's a great privilege to come and a great pleasure to visit this beautiful country. So my task is to talk a little about the Australian experience, and I think, like many people, we would regard this as a bit of a cyclic process. Obviously, when we're talking about the use of bacteriophages therapeutically, to which we've had a great introduction this morning, so I'm not going to talk about how they work or all that sort of stuff, but I will talk about our experience. We start, like many people, with a collection of bacteriophages, and sometimes you'll be asked to find something that you don't have an answer for, and some of these requests can be quite obscure. So one of the challenges, of course, for clinicians who produce phages for therapeutic use is to determine whether they can provide an answer to every question, because this is a truly precise therapy. And because it is a precision therapy, it is very important that it be based on good quality understanding of the relevant genomic epidemiology, which needs to be built around surveillance structures. And that's a very important underpinning to the whole process. Once you have that established, of course, like all clinical enterprises and especially clinical trials, it's all about patient selection. And that requires a fair bit of clinical input and clinical specialist knowledge to determine who will profit most from such an intervention. And of course it requires an education process of people who are unfamiliar with the use of bacteriophages, because they are sometimes perceived by clinical staff who are unused to them as potentially problematic, in the same way that genetically modified therapeutics are sometimes perceived as potentially problematic. So if you have, for example, pregnant nurses, they might be anxious about the use of bacteriophages. So there's certainly an education process required. Once you feel like you have got an established process, then there is the conventional laboratory side, obviously matching the right predator to the prey, so matching the right phage to the bacterial target and producing it cleanly. A lot of the work we do, and I think you'll hear more about this afternoon, is heavily dependent on an understanding of the genomic epidemiology of the targets, that is the bacterial prey you're trying to treat, and we use genomics tools for a lot of our production processes, including in-process monitoring. The role of patient treatment and monitoring is something that's worth discussing, but essentially we've taken the view that because we are learning as we go, we need to be monitoring fairly intensively. So we spend a lot of time and energy understanding the patient process as the therapy evolves. And finally, of course, it's important to provide detailed analyses of patient cases, because all our clinical colleagues are watching to see what's going on with this. Is this something we should be embracing, or is this just another crackpot scheme that's not going to go anywhere? When we went to set all this up, our intention was to gather in a small country, gather all the experts in the country around all the different universities, and you can see that includes one of our speakers, Jeremy, here in the audience, to get hold of the scientific expertise and the clinical expertise that you need to start an enterprise like this, and more importantly, to carry people with you who would be perceived as relevant knowledge leaders. That's very important, I think, or you don't get any traction. And clearly, as we set out to do this, it was important for us to partner with international colleagues who have expertise, clinical and particularly scientific expertise. And again, one of our speakers was amongst our generous international colleagues who supported us to try and produce therapeutics for human use. When we took this program, Jeremy and I were involved with a lot of our colleagues around the country in a rather ambitious attempt to get funding to set this up in the country. And the review of that program said, perhaps unsurprisingly, given that we'd got everyone together, that the scientific plan was fine, the clinical plan was fine, but when we met the venture capitalists who were reviewing this process, and they asked, how are we going to make this economically viable? That's where things fell flat. And the things that were raised by people who want to invest in this and want to understand whether this is a cost-effective approach were the regulatory framework. This is a high-risk area. So clearly, if you are going to develop phage therapy in a nation and you do not have a regulatory framework to operate in, that constitutes enormous risk, because an investor who says, I want to set up a company to make phages, might find that the government says, well, I'm sorry, you can't use them for the purpose you thought you were going to use them for. So the regulatory framework's very important to resolve. I'm not going to talk about this at length, but it is a big issue and a lot of countries are wrestling with it right now, and I hope that we will get some clarity about regulatory frameworks in various jurisdictions within the next 2 years, or maybe even less. Similarly, and partly for that reason, the market is not clear. So do we just use phages to rescue people in extremis? Do we just use it for antibiotic resistance? Do we use it in spaces where antibiotics are not used at the moment because we're concerned about driving antibiotic resistance, and so they counter antimicrobial stewardship principles. This has enormous impact on the perceived markets and therefore on the business opportunities for people who might want to support a major enterprise such as antibiotic production, or in this case, phage production. And the other thing that is raised commonly is that if you can get a bunch of kids from high school or university, as we heard from one of our previous speakers, to go out, put a bucket in the water and come back and with hardly any support raise bacteriophages that could potentially be used therapeutically. How on earth does a company that wants to make this economically viable obtain the monopoly position that they require? And so if you look around the world at people trying to do phage therapy, there's a rush to genetically modify them, to capture some sort of a product that they make and clone it, to wrap some sort of a patenting process around that, to make it commercially viable. And these are all relevant because one of the key questions for government is, do we have to do this, or can we just let the private sector manage it? So one of the questions we asked, and we did this by asking professional health economists, so this was simply a subcontracted process. We simply asked the question, what would happen if in 1 year, and we chose 2018 because we had great data from that, in one particular year we made available phage therapy for the treatment of infected hips and knees, just infected hips and knees. And we asked them to calculate the potential savings that would have occurred with a couple of key assumptions. Now, the input assumptions were that we were paying around about 24,000 Australian dollars per course. Now, the costs, very difficult to calculate the costings of these things, and we could talk about that perhaps at another time, but that was the input assumption. The other input assumption was 55% efficacy, and that is in people who had already failed a DARE procedure or a two-stage procedure to rescue an infected hip or knee. So these were high-risk people. And in fact, that 55% efficacy, I think if you look at the published literature now, would be regarded as an underestimate. But they were the input assumptions, and the conclusions were that in a country of 25 million people, we would have saved nearly 2,000 operations, more than 300 premature deaths, and 123 million Australian dollars. Just in 1 year. If you compare this with other models where you might employ this, we put up cystic fibrosis because around the world this is the early adopter community, particularly in first world countries. And I think we're seeing in the US and in Australia the uptake of anti-pseudomonal treatment in the cystic fibrosis community is one of the early areas. And the benefit-cost ratio that's calculated for that, in other words, this is how much we spend, these are the long-term savings, was about 3 and a bit. So in other words, for every dollar you put in, every ringgit you put in, you get more than 3 back. If you look at prosthetic joint infections, which is the one I mentioned a second ago, the estimate is over $17 saved for every dollar of input. And if you look at severe sepsis, because the other model we put up was bone marrow transplantation, neutropenic sepsis, because people often talk about that as a rescue strategy, and again, about $3.50 saving for every dollar that you put in. And if you look at renal transplantation as a model, the published benefit-cost ratio for a kidney transplant, which is accepted as standard of care, is about 5.5. So this compares favourably with renal transplantation, which is not privately funded. This is a public good. So it's up there in the category of things that you would regard as important in public sector health. Clearly, there is a distinction to be made around economies of scale. So that if you have every single treatment just used once, so every patient gets a fresh batch of phages made for them, then that is more expensive than having one batch of phages made that 100 people can benefit from. So there are clear benefits to be had in economies of scale. If you look at those figures I said before, a net present benefit overall for the 3 phage therapy models that we put up a second ago was about 900,000 ringgits per patient. So that's the benefit for the therapy, and I mentioned the overall cost-benefit ratio. So one of the questions we're asking now is what price is acceptable. In Australian and in UK hospitals, the price that we would pay for a quality-adjusted life year, none of this is published, but you can infer it by looking back at what governments support, is around about the 35,000 Australian dollar mark, and that is well within what we would regard phage therapy. So it's clearly in that public good space, that clear public benefit. And the question remains then, how do we do this safely? So coming back to this question just briefly about the economies of scale, if we make a big batch of phages or a monstrous batch of phages, the actual labour costs and the biotechnology inputs are very similar. So there are clear benefits of scale from making a small batch versus a huge batch, as long as you don't waste the huge batch. A lot of the therapy we do is one-offs. We don't treat a lot of people with it, so we throw most of a batch out. And interestingly, I was assuming that just making buckets and buckets of phages, if enough people would want it, would make a big difference. But in fact, a lot of it, at least for us, are the matching and diagnostic costs. So the economies of scale will be harvested better, I think, after the clinical trials are in and it's clear how to use them. At the moment, the clinical trial is a classic example of an efficient way to use it. You probably only get 50% wastage or maybe 70% wastage in a clinical trial. And so the nearest approach we've got to reach the economies of scale is to understand the genomic epidemiology of a particular problem well enough to be able to construct a sort of a consensus therapy with multiple phages in a so-called cocktail. And Belinton Jeremy will talk to you more about that a bit later. So our case mix, we supply all around the country, and there are therapy centres all around the country now. And our main requests are Pseudomonas, Staph aureus, the multi-resistant gram-negatives, and Mycobacterium abscessus. And the early adopters we've talked about before, the critical care doctors, the transplant doctors, the cystic fibrosis community, and of course I've talked a little bit about bone and joint. And it's relatively straightforward to ship things around. Modern aircraft, you can get something in 24 hours. So we send phages routinely a greater distance than London to Moscow, and it gets there overnight and gets there in good condition. So that's not really a logistic challenge. I'm not going to talk about cold chain, but I don't think it's a major problem. We did ask our infectious diseases community in Australia what they thought about all this. And they said, basically, we think the story is that this stuff is pretty safe. We've already heard that this morning. We would like to see timeliness in its availability, and we want to see structure about its use. And the bottom word picture is the highlights of what Australians thought were important to target. And you can see the gram-negatives, the non-tuberculous mycobacteria, prosthetic joint infections feature strongly. A similar survey, so this was the same survey instrument, was done in Korea as well, a different environment. And they had much the same kind of questions, but were less sanguine about the safety data. So it's about how familiar your clinical community is with this, how confident they feel about the safety data that are available at the moment, because that's a key determinant, I think, of uptake. And similarly, the targeting question, again, unsurprisingly, I think here, unsurprisingly in Korea, they were more interested in Acinetobacter, because it's more of a problem there than in Australia. So where are we with clinical trials? So this is a little out of date. This is taken from the clintrials.gov. So we have a lot of trials going on in the GI space, a couple in bone and joint. You can see them listed there. The ones that we have a little bit of familiarity with in Australia are the trials for chest infections. We're running one locally. We've just recently conducted or been part of a US-based study in staph bacteremia, and there are a few all-comers trials, and ours is one of them, and I'll talk a little bit about that because it might be useful. One that's coming, which is an Australian trial that is a little bit multinational now, and that's a Staph aureus adaptive platform trial. And in fact, adaptive platform models are probably a good way, I think, to test adjunctive phage therapy. What have we learnt from clinical cases though? Because in Australia we set out to try and answer the problem of people who had nowhere to go. So we've now treated about 50 odd patients, 32 in our latest iteration of the STAMP protocol. Overall, the efficacy in the face of failed therapy is more than 75%, but look, I'm not going to talk about efficacy because it's uncontrolled data and because it was all about patient selection. So any clinician will tell you they can determine the outcome of a trial if you let them choose the patients that go into it. So I don't think the efficacy signal is very useful, but it does at least give us some assurance that most of the times, and remember the input strategy for us is people who have already failed everything, medical and surgical therapy. I'll talk about that in a second, but we've learnt that it appears to work quite well. We get occasional inflammatory responses that for clinicians who are familiar with treatment of syphilis and tuberculosis, it's like a Jarisch-Herxheimer response. And we see it in about 1 in 20 patients that we treat. It's usually not very severe. It's probably no worse than a vancomycin infusion reaction usually. Neutralising antibodies are common, but not universal, and they vary probably with the virus. There's a little bit of uncertainty about this, but it's certainly true that we get, for example, with the K viruses, which are commonly used against Staph aureus, we get almost universal neutralising antibodies evolving at the exact same time that you would normally expect neutralising antibodies to rise after you give an infusion of a virus. So the sort of time frame is typically 7 to 10 days. You can see it in the transcriptome, you can see it in the neutralising antibodies responses in the blood, but if you don't treat beyond that second week or two, it's not going to be relevant at all, I think. We use it intravenous. We use it through every route that you could use an antibiotic. So topical, intravenous, intercavities, intravesical, oral, whatever you like. Nebulized and IV is common. Our main ambition when we set therapy up is to try and create an environment in which the bacteria are simply unable to adapt to their ancient foe. And I think we'll hear more about this in subsequent talks. This is one of our early experiences where we looked at bloodstream infection. So this was a standard combination of 3 K viruses, and for those who are unfamiliar, these are really common viruses that are used as obligate predators of Staph aureus. And the entry criteria was severe, prolonged sepsis due to Staph aureus infection. They were all intensive care patients. They were almost all on life support, and most of them had endocarditis, so very severe disease. And what we learnt from this is published, so I don't want to spend much time on it, but what we learnt that we did not see resistance despite high bacterial and viral loads. So those top bars show the presence of positive blood cultures, and the bottom bars show the duration of the phage therapy you can see, and those big spikes you see are bacterial counts in the bloodstream. So we had culture positive, high counts of bacteria, large amounts of virus, so 10 to the 9 plaque-forming units, so there are 10 to the 9 viruses injected into the vein at the same time, but we did not see resistance emerging. And that tells us something about how things work. We mentioned the antibodies, that was common. The other thing I mentioned was this inflammatory response, which is really quite common, but for the clinicians in the audience, I think you would find this unsurprising, because we're used to the notion that if you have a large burden of disease that has managed to hide in a little bit of a protected space, like we see with syphilis and tuberculosis, most notoriously, then that inflammatory response is so predictable Then in cases of TB, for example, if you have TB in brain or pericarditis, for example, we would always use steroids in advance of with the therapy, because the inflammatory response is so predictable. So you do see this, you get a classic human transcriptome response. Here we see example where on the vertical axis we have the presence of the bacterial burden in the bloodstream, which are those orange stripes, and this was treating a Pseudomonas infection in a little girl who had an infected leg, an osteomyelitis in her leg. And you can see that after those doses, which are those vertical arrows at the bottom, around about day 8 you start to see this big response of virus that you can find in the bloodstream. So it's common that once you get that amplification response, you can detect it in tissue fluids. Now, of course, if you're treating a bone or a wound or another site, you might not get so much spill into the bloodstream, which is the easy access. But the amplification response, which we heard before from Siva talking about this morning, is quite detectable in human tissue fluids. So what do? We use this phage therapy approach, which we call the standardised therapy and monitoring protocol. And our rationale was that we're treating patients with compassionate use phages, but we needed a way to monitor what was going on, and that had to be done as a clinical trial. So that involves standardised sampling. We recommend standard dosing, so that most of our patients are getting 1,000 million viruses injected into the blood twice a day, usually for 2 weeks, and we monitor in a very standardised way what the viruses are doing, what the bacteria are doing, and what the human's doing, because there's 3 players in this, and we need to understand them all to monitor progress. So I think the rationale behind this was to not only learn as we go, because despite our long history with phage therapy, we still don't really fully understand what we're doing, and to also understand really what it costs to do it. And we've now treated more than 125 patients. I've updated this slide a little bit, which is why we've got numbers at the bottom. And of the patients that come for requests, and this is, remember I talked before about patient selection, we would triage about 100 of them or thereabouts to receive therapy. 80% of those then get treated, a few clinically progress, because we can't produce phages the same day like an antibiotic at a pharmacy. It usually takes, in our experience, at least 5 days, but typically a month, 2 months, to produce a clean therapeutic phage suitable for IV injection. So 80% of them get treated, which means we make a lot of our own batches now, and we're scaling that up, so we're looking for those efficiencies of scale. And we've treated, as I said, a total of about 50-odd patients now. So what have we learned? I mentioned that the efficacy is a bit of an unknown, but most of our patients are getting better. We see inflammatory responses occasionally. Neutralising antibodies, we think, probably varies with the virus you choose, but certainly true for some. And you can detect these amplification responses to tell that you've at least got the predator to meet the prey in most people. I'm going to close now. I'm not sure how I am for time. I haven't been keeping an eye on it, because this is just about my last 2 or 3 slides now. So I'm just looking for a nod. I'm fine, okay. So I just wanted to give an illustration to kind of set up some of the talks we're having this afternoon. So this is one of our most recent cases. This is a 16-year-old boy with acute leukemia. Many of the clinicians will be familiar with this kind of story. So failed treatment for ALL, getting set for a bone marrow transplant, 2nd round of chemotherapy, allosparaginase-induced necrotizing pancreatitis, a dreadful complication for anyone. And by the time we met this young fellow, he'd spent more than 200 days in ICU, most of that on life support. And he'd had a lot of antibiotics, and he just had one dominant pathogen selected. And this was a Klebsiella aerogenes, which had a common resistance gene, one of the IMP genes. And so it was really difficult to treat. we gave him we prepared some local therapeutic phages and gave him a couple of courses, but we monitored his therapy and his bacterial populations as we went, and we could see it changing. And in fact, by the time we got towards the end of his therapy, we did not have a phage in our stock that was going to do the job. So we reached out to Balint, and he was able to produce a nice couple of phages, which we then administered the coup de grace on that bacterial population with. Now, by this stage, these bacteria were really quite diversified, really quite evolved, and the reason I'm making this point is because in high burden populations, as we often get served up, you have to understand the diversification in that population if you're going to manage it. If you don't, there is a high risk of therapeutic failure. Now, it may be able to overcome that with a various number of strategies, which you might hear about this afternoon, but basically you have to bear in mind that our fundamental job here is to try and make it so difficult for the bacteria to adapt to all the things they're dealing with: complement, iron starvation, antibiotics, bacteriophages, oxidative stress, all those things, and basically make it impossible for them to continue. And that was a very successful story. I think the other point to make, we haven't published these data, but it will be very reminiscent for those who are familiar with the Patterson case, which Jeremy was involved in which exactly the same scenario. You had a high burden of disease, you had massive diversification in the population, and you had to chase the dominant bacteria with multiple different phages because you're trying to manage its adaptive landscape. And that was famously successful. So on reflection, what were the things that I think made the difference to getting things up and running, and what would I look to as most important points? There's no doubt that we need clinical uptake, we need clinical leadership. One of our biggest challenges has been to convince practicing infectious diseases specialists that this is safe and this is potentially effective. We have had a lot of discussions, and this is something that needs to be considered by every country, about scope of practice. So, for example, should you make this available to anyone who wishes to prescribe, or should you restrict it to expert prescribers and even provide training? I would argue the latter is appropriate. The regulatory framework we've talked about a little bit. The funding model is a challenge. We've also mentioned that, but I think we should remember that one of the key points here is about sovereign capacity, which I think we've heard alluded to this morning. I think that's very important for every country, certainly for our country. And there's no doubt that there is enormous power in government leadership in this space at the moment. So if you have something that you perceive to be a national good and you have convincing economic analyses that make sense to you, then the question is how best to proceed from there. So if I was going to redo this in Australia, or if I was to make any suggestions for anyone else, I would say you need to do your local health technology analysis. You need professional health economists to look at this, look at the data, look at your populations and answer the questions about whether this is good and how good it's likely to be. You need to manage the regulatory frameworks. That's very important. It may be that your Ministry of Health or your government could conduct a strategy in which you can consider regulatory frameworks that are fit for purpose for this sort of thing. And you would need to consider whether you simply wait for external production or whether you make your own, and that's that same sovereign risk question. I think it's an important point to make that there is no, and we heard this great discussion about the CFAGE program, beautiful example of how you can leverage academic expertise. So you make huge investments as a government in your academic leadership in your country, your doctors and your scientists. And the logical thing, if you're dealing with something new and difficult, is to leverage that investment. So I would consider that a strong academic base is essential, and it's a cost-effective way to proceed with this sort of thing. And you can set up networks and centres just through simple tendering processes, and the academic environment is used to this. And to actually then kick things off, again, I'd come back to my clinical leadership thing. I think if you get the senior clinicians all with a consensus position about how this should be done and how they're going to treat their patients, I think that is a logical way to start. In our country now, we've got so many people who are prescribing that we need to set up a clinical training course. So we'll be starting that this year, where we have a simple training course in the way that we train our general practitioners to prescribe for HIV therapy. So in our country, HIV therapy is the province of the infectious disease specialists and the immunologists, but you can do a training course for any prescriber. And so that concept of prescriber training is well established, and I think it's a model that we'll probably look to. And I'm going to stop there and see if there's any questions. So thank you very much for your attention.
Is there any question from the audience? No question, Prof. Thank you so much, Professor Jonathan, for sharing with us how phage therapy system is being implemented in Australia and how we can start in Malaysia. Next, let us continue the symposium with a talk on using phages to make antibiotic resistance abnormally sensitive to antibiotics again, phage production for personalized therapy. Our speaker, Associate Professor Dr. Jeremy Barr, is an expert in bacteriophage research and phage therapy. Currently based at Monash University, Associate Professor Dr. Jeremy leads research on phage bacteria interactions and directs the Monash Phage Foundry, focusing on phage production for antimicrobial resistance. Recognized with an NIH Investigator Fellowship in 2024, his translational work has made significant strides in combating AMR. Let us warmly welcome Associate Professor Dr. Jeremy to share his insights. Please welcome.
Thank you. Thank you for the invitation. Thank you, Toby, Phages for Global Health, Ministry of Health Malaysia, for the invitation to come out to speak. Great to be here. Great to see so many friends and colleagues as well. So my name is Jeremy Barr, and I'm currently an Associate Professor at Monash University and School of Biological Sciences. And I'm going to tell you a little bit of a different story. I'm really going to take you into a bit more of a deep dive into some of the fundamental biology of this phage-host interaction. I'm going to focus on Acinetobacter baumannii. This is a really critical clinical pathogen. We heard a great talk this morning on its burden of disease, and understand it's one of the sort of early potential case studies or case pathogens that you're looking to implement here in Malaysia. So I'm going to try to answer this question, and that is phage therapy an effective treatment for Acinetobacter baumannii? I think before we can even begin to answer this, we really need to understand a little bit about this pathogen, or a lot about this pathogen. We know that crab in particular has a high burden of morbidity and mortality, and in 2019 it was predicted to be associated with nearly 500,000 deaths globally. It is a diverse pathogen with extensive antimicrobial resistance and virulence determinants. And one of the really unique, somewhat unique aspects of the biology of Acinetobacter baumannii is it produces this bacterial capsule. This component of these capsular polysaccharides, these short repeating sugar units that are secreted and cross-linked and form this sort of protective layer or sugar shell that coats the external surfaces of this pathogen. And this capsule is produced by a cluster of about 20 to 25 genes that we call the K locus, and we can also define these on their KL type. It's these cluster of genes that are responsible for the production, synthesis, cross-linking, and export of these complex polysaccharides. And as a result, you see a huge diversity in the capsule structure in A. baumannii. So here you're seeing just some colony morphology images, and you can see that these look quite diverse. And if we look at these isolates under the electron microscope, you can see the diversity in the capsule productions with sort of very dense, hypersecreted, to even lower amounts of capsule production. And to date, we've been able to classify these KL genes based on their KL type, based on the genomic orientation and grouping of these genes. And there's over 230 KL types, and this number is continuing to increase as we continue to sample and sequence this pathogen. And so I want to share this. This is a snapshot of our phage host library from our lab. So I'm showing you up the top, we've got a range of clinical Acinetobacter baumannii isolates. Several of these we've identified the KL types. You probably can't read them, but they're color-coded in that second row. On the left-hand side is a range of virulent phages that we've isolated and characterized in our lab. And the heat map here, so an orange square shows that that phage can infect and kill that Acinetobacter baumannii isolate. And the point that I'm making here is that for the majority of these phages, they're highly specific. They're only infecting a single strain, and where that phage appears to have broader host range, it's really refined and only really infecting within its KL type. So this KL type is really the major determinant for phage infectivity in A. baumannii, and we typically have to match our phages to the capsule locus and those complex sugars. And this makes it extremely difficult to find the correct phage for your correct A. baumannii pathogen. Okay, so with that sort of broad primer and introduction to the pathogen, I'm actually going to tell you two stories today. These are both research stories from some of our students. The first is from Fernando, who's a PhD graduate. He's now working at a postdoc at the Alfred Hospital and leading sort of clinical phage therapy work, and Leo Khan, who's a third-year PhD student in our lab. And really the key point here I'm going to focus on is that third point. And in my opinion, we really need to understand the fundamental biology of the phages and the pathogens if we want to implement successful and consistent phage therapy. I think we know that yes, phages will kill a pathogen and we can use that clinically, but I believe that we can and we should do better by leveraging this fundamental research. So the first story I'm going to tell you is looking at this phage here. So this is phage FGO2, and it infects the clinical A. baumannii strain AB900 that I believe is a KL22 isolate, and you can see its TM morphology and its plaque morphology on this multi-drug resistant clinical strain. I'm going to show you several growth curves in this talk. So we've got here AB900 growing on its own, fairly typical growth curve where you see increase in optical density over time. If we grow this pathogen in the presence of a clinically relevant concentration of imipenem, we see it slows the bacterial growth, but it doesn't really affect its maximal OD. Yet if we grow this in the presence of phage FGO2, we get excellent bacterial killing and suppression. However, if we extend this growth curve out, we eventually see the emergence of phage resistance. This was touched on in some of the earlier talks. This is a common phenomenon, particularly in the lab. You must almost always see the emergence of phage-resistant mutants under lab strain conditions. It does happen in vivo to a lesser extent. So we really wanted to use this as our starting point. How is this bacterium evolving to become resistant to this phage? And could we use that information to identify the bacterial receptor? So again, this is this structure, the surface component that the bacteriophage utilizes to mediate that infection. So to do this, we isolated those phage-resistant mutants. So I'm showing you two bacterial hosts growing in the presence of the phage, the wild type in red, and you can see phage killing, and a completely phage-resistant mutant that we've isolated in blue, again growing in the presence of that phage, and you can see it's completely immune. It's invisible. It does not care whatsoever about that phage. And we started to characterize these phage-resistant mutants phenotypically. We looked at their scanning electron microscopy, their surface structure. We did a manifold capsule strain down the bottom, and you can see that the phage-resistant mutants appear to have lost that capsule structure, and that was recapitulated in that capsule strain. And we see the loss of that counter strain sorry, I've just lost my screen up here. With loss of that capsule counter strain. So we then sent those receptors, or sent those isolates out for sequencing. We do whole genome sequencing, and we're able to do a pairwise comparison between the wild type and the phage-resistant mutant, and we found a single causative mutation within a gene known as GPI6. Here we had a single nucleotide deletion that led to an early stop codon, leading to a putative loss of function in that gene. And perhaps not surprisingly, this gene is a glucose phosphate isomerase, or it's associated with changing or addition of a sugar onto a growing capsule chain, and it's involved in the K locus. So it's a critical gene in the production and synthesis of those capsular polysaccharides. So we had a putative link that suggesting that the capsule was the receptor. So we wanted to go and validate that with molecular mechanisms. We generated some knockout mutants. This strain was thankfully sensitive to kanamycin, was one of the only antibiotics that it was sensitive to. So we took that kanamycin resistance gene, we flipped it into that GPI6 gene, thereby knocking it out, and we're able to use that negative selection to isolate those knockout mutants. And we did this phage absorption assay, and I'll talk you through this experiment in a little bit more detail. So here, let me see if I can get this pointer working. I'll just explain this. So on the y-axis, you're measuring the absorption of free phage, so the loss of free phage as they go and infect their bacterial host. In dark red, we've got the wild-type host, and you can see that that wild-type host is absorbing about 1 to 2 logs of free phage over about a 20-minute period as the phages are infecting and entering into that bacterial cell. In dark blue, we've got our in vitro phage-resistant mutant, and this does not absorb any phages. It's completely invisible and immune to that phage. If we knock out the GPI6 gene in light blue, we can recapitulate that phage-resistant phenotype and we have no phage absorption. And then if we complement that gene back into that knockout mutant in that light red color, we can restore phage infectivity, thereby validating that yes, for this phage, it is mediating infection via the bacterial capsule. So this loss of bacterial capsule allows the host to become phage resistant, but that capsule has a very important function. It has very important virulence, determinants, and other physiological functions. So becoming phage resistant comes at a fitness cost. And we found in this phage host pair, this led to an increase in killing in the presence of human complement through these serum assays. We saw about a 5 to 6 log increase in bacterial killing. We found expanded phage infectivity to other phages in our collection. And perhaps most interestingly, we found examples of antibiotic resensitization, particularly to beta-lactams, and with ceftaz was our most highest responding antibiotic. We actually saw a change from clinically resistant to clinically sensitive phenotype upon losing that capsule and becoming phage resistant. So a brief sort of recap here on this component. So FGO2 uses the bacterial receptor as its, capsule as its receptor. This leads to the repeatable emergence of phage-resistant mutations that have loss-of-function mutations in those capsule-producing genes, and this capsule is a major virulence factor. So losing this capsule allows it to become phage-resistant, but this comes at a fitness cost. And we found that these phage-resistant capsule-deficient mutants have increased killing to human complement, additional phages, and were resensitized to beta-lactam antibiotics. So this was published a couple of years ago in Nature Microbe. And so this concept of understanding and leveraging this fundamental biology is that we can improve clinical outcomes. So we can take phage therapy, we can treat antibiotic-resistant pathogens, but by understanding those mechanisms, the hope is that we can extend the clinical outcome. And in this example, this is in the use of these repotentiated antibiotics. So this is a great concept, but does it work? And this is what we wanted to test next. So we moved forward with a preclinical trial. We designed 4 treatment groups. The first group of mice received a PBS control. The second group just received that ceftaz antibiotic. The third group received phage FGO2 on its own, and the fourth received a combination of phage plus antibiotic. This was a really acute infection model, so mice were given a lethal dose of this A-Balmane clinical host intraperitoneally. 1 hour later, they received their respective treatment. 8 hours later, every treatment group that received an antibiotic received a second dose due to its half-life, but no additional phages were given, and this experiment ended at 11 hours. And so I'm showing here the reduction in bacterial load across all of the mice with each of those increasing antimicrobial conditions. And so you can see that we have a continued decrease in bacterial colonization. However, we didn't see a significant difference between our phage-only and our combination phage antibiotic treatment group. And the reason for this was that we had to end this experiment early. Our control and antibiotic-only mice were incredibly sick, really high bacterial loads, approaching 10 to the 10 CFU per gram of tissue, and we hit our ethical endpoints and we couldn't take this experiment any further. And that simply wasn't long enough for us to select for the emergence of phage resistance in vivo and then counter-select for this with those antibiotics. So we designed a second stage of this trial. Here we just compared the phage only to the phage antibiotic combination, the exact same experimental setup, and this experiment went for the full 16 hours. And here we were able to show a significant reduction in bacterial colonization with that phage antibiotic combination treatment. We had phage replication, and we had significant emergence of phage resistance in all of those in vivo isolates. And so summarizing this part of the talk, we're proposing this as a sort of 1-2 punch strategy where this phage targets the A-Balmane capsule, it drives the consistent emergence of phage-resistant mutants via loss-of-function mutations in those capsule-producing genes. As a result, these mutants are less virulent and they're resensitized to beta-lactam antibiotics. And again, the point here is understanding that biology is that we can hopefully leverage these clinical trade-offs and improve the efficacy of phage therapy in the future. So that work is all published, and so I want to pivot into a new study in our lab. Again, this is looking at Acinetobacter baumannii. This work's done by Leo Khan, who's a third-year PhD student, and Leo's been studying this phage that he's named ABS1, short for Acinetobacter baumannii Slayer 1, which is great. I always encourage these creative names. Beautiful-looking phage TEM and SEM structures. And we specifically started looking at this ABS1 because it had a really unique host range map. And so I told you at the start of the talk that phages almost exclusively infect within their KL type. And this phage was unique as it actually infected across four diverse KL types that had different polysaccharide components. Still, this is quite a small host range. It's only 5% of clinically relevant KL types, or in other words, it's still not affecting over 95% of Acinetobacter baumannii global prevalent KL strains. Nevertheless, we continued to investigate this. We generated a whole genome sequence of this. It's got about 100 kb genome, and it's a member of the Sackley virus genus. But when we started looking to in vitro, it had some really unique phenotypes. So here we've got the wild-type KL83 host growing on its own, and in green, the host alongside this ABS1. And this is a highly efficient virulent phage. It was actually able to completely suppress this bacterial host. We did not see the emergence of phage resistant under in vitro conditions. And in fact, we did extensive CFU testing, and this phage is actually eradicating this bacterial host in vitro. We cannot recover viable CFUs in lab strain conditions, which is very, very unique for this phage host pathogen. And just as a recap, this was our understanding, right? The majority of these phages against these pathogens are binding the capsule, and that's how they're infecting. And by becoming phage resistant, losing the capsule, the phage is no longer able to infect. And so we generated a phage resistant mutant, but we found that upon becoming phage resistant, this phage resistant mutant actually tripled the amount of capsule it was producing. And so this really gave us pause, and so we had to go back and rethink the mechanisms here. And so we came up with an alternative hypothesis that rather than recognizing the capsule, this phage-resistant variant was hypersecreting the capsule, and it was sort of pushing out and excluding the phage from infecting through capsule overproduction. And so the alternative hypothesis here is that this phage was a non-capsule targeting phage. It was targeting some other structure on the bacterial cell surface. So we actually spent a lot of time trying to isolate a phage-resistant mutant to validate what this receptor is. We literally spent nearly 9 to 12 months, and the issue was that there's a really high fitness cost of generating a phage-resistant mutant. Many times we'd isolate a mutant, we'd sequence it, and it would revert back to wild type. Eventually, through this sort of multi-pronged approach, we were able to identify the receptor, and we identified this as a BTUB3. This is a conserved TON-B-dependent receptor that's putatively associated with copper transport across the bacterial cell membrane. And this is a really telling experiment. So I'm going to show you a couple growths here. So this is a wild-type KL2 strain. This is one of the most predominant KL types globally for infection. And this is grown in the presence of ABS1. And so you can see this ABS1 phage cannot infect this KL2 strain. There's no infectivity. There's no killing against this capsule-producing KL2 strain. We then knocked out the BTUB receptor in this strain, and we see that there's a fitness cost. There's a reduction in bacterial growth. This appears to be a very important receptor for the growth of this pathogen, but we don't have phage infection in this mutant. Yet when we knocked out the capsule-producing genes in this strain, we saw complete eradication of this bacterial host from this ABS1. And when we did the double knockout, knocking out both the capsule-producing genes and the BTUB receptor, again, that phage is no longer able to infect, and we see it cannot kill that bacterial host. So this was a really telling experiment and sort of confirmed our infection mechanism. And so what we're sort of proposing here is that this ABS1 may be a sort of universal donor that we can add alongside other capsule-degrading agents. And of course, the first one that we tried are capsule-targeting phages. So here I'm going to show you another growth curve. Here we've got a different KL22. This is sort of the third most prevalent KL type globally. Infecting this with ABS1 alone, it cannot infect this capsule, and there's no infection. If we target this with a capsule-targeting phage, we see this capsule-targeting phage is able to kill the bacterial host, and then we see the emergence of phage-resistant mutants, likely through that loss of capsule production, and then this allows this secondary killing of ABS1 to come in, and we get extremely good suppression and killing with this double phage combination. But we can also take this further. We can use depolymerases. These are phage-encoded enzymes that specifically attack and degrade the capsule polysaccharides to strip back and remove those agents, thereby allowing ABS1 infectivity. And again, a similar in vitro experiment with another KL type, KL14. ABS1 cannot infect this capsulated strain. We can add a depolymerase to remove and reduce those capsular polysaccharides. There is a slight reduction in bacterial growth, yet when we combine the two, we see a significant reduction. And I believe this outgrowth at the end here is likely due to the quenching of that depolymerase as it's eventually used up, being an enzymatic reaction. So what we're proposing is that this ABS1 can be really a broad host range phage if we can combine it with mechanisms to bypass that bacterial capsule. So ABS1 can naturally infect 4 of these strains. We actually predict that these are under capsule producing strains, allowing it to get activity. And we've now been able to show that ABS1 can actually infect and kill the 3 most predominant KL clades, KL2, 3, and 22. We've done some preliminary bioinformatic analysis showing that this BTUB receptor appears to be present in more than 90 to 95% of clinical A. baumannii isolates. So still ongoing work, but really promising to improve the clinical utility of these phages. So I'm going to come to this question, and just looking at how I'm doing on time. Is phage therapy an effective treatment for A. baumannii? I think the answer is yes, but this is a really challenging host pathogen to focus on. It probably wouldn't be my first recommendation. However, that's a little bit hypocritical of me because this is also where I started our phage therapy work. So I think some broad conclusions that I would make is that yes, I think we do need to understand the fundamental biology of the phage and the host that we're using and that we're targeting. This pathogen in particular is highly diverse, particularly on its capsule loci, and I hope I illustrated that, and the majority of phages that infect it are very, very specific for those capsule types. And based on this, I think it's going to be unlikely that we're going to be able to develop broad spectrum phage cocktails against this against this isolate. And I know Balint's probably going to contradict me in a couple half an hour's time with his talk, but I think a better approach is really a personalized approach, leveraging and understanding the diversity of the capsule types in your local country and building phage collections. And I know that's what Balint's really going to speak on, so I'm excited to hear his talk. And really targeted phage isolation against those clinically emergent strains and matching that phage infectivity. Now, I realize I'm almost out of time, so I'm not going to spend too long on here, but I wanted to just give a really brief overview snapshot of our local phage manufacturing production. This is really in collaboration with Phage Australia and John and the team up at Westmead that we've been building this National Institute to sort of establish decentralized phage manufacturing that's allowing us to treat patients via compassionate use in lots of decentralized sites across Australia. Our goal for our production was to have an efficient, scalable, reproducible, and safe production pipeline. Important, this is not a GMP facility. I know Frank's going to speak a lot on this later. This is very, very much for compassionate use. So this is sort of an overview of our sort of 5-step process that we use to produce clinical grade phages for compassionate use. We begin in our microbiology lab. We produce typically several liters of the bacterial host. This is usually the patient's clinical pathogen, and we allow the phage that can infect that pathogen to grow up to quite high titer. We then move this through a custom designed depth and sterilization filtration pathway. We don't use centrifugation here, and this step is completely scalable. We can use larger size filters. This can work with a 50 ml product up to a sort of 50 liter product, and we can take raw bacterial lysates, filter those first through depth, then sterilizing grade filtrations, and at that point we have a filter sterilized bulk phage lysate. We then take that from our microbiology research labs into a clean room. It's not a clean facility in any means, but we make an effort not to bring any bacterial host and try to have a higher cleanliness and protection of those samples. We then move into a buffer replacement and washing. We've got partnerships with Cytiva in use of their equipment, so using TFF processes to first dilute our product, then wash and concentrate those down into a buffer of choice. We typically concentrate down to about 200 mils of product. The fourth panel here is still a bit of a wish list. We've got a couple of grants to try to get some additional equipment funding. love to set up a chromatography system that we could semi-automate this process for the removal of bacterial endotoxins. This is really the most challenging and time-consuming step in this whole protocol. At the moment, we're using a combination of octanol chemical treatments along with some commercial kits, but we really have to adapt those based on every patient and pathogen that we're treating. This platform here would allow this to be somewhat standardized and improve the throughput. And finally, we package all of our products into multi-use syringe vials. We're moving towards a sort of 10-dose vial system. We produce multiple vials per batch, and that entire batch is sent out for third-party validation for sterility and endotoxin, and then we pass all of those over to the clinical team at the Alfred, who lead the clinical translational arm of this work. Happy to talk about this in a bit more detail later if people are interested, but I'll wrap up. Big thanks to Fernando, Dinesh and Leo for doing a lot of the research, our collaborators, the Peleg Lab, Alfred Hospital, Phage Australia, the Centre to Impact AMR, and our funders. And thank you all for listening. Cheers.
Thank you, Associate Professor Dr. Jeremy, for his insights, mostly on Acinetobacter baumannii. Ladies and gentlemen, our next talk is from closer to home, Singapore. Our speaker, Associate Professor Dr. Kwa Lei Hoon Andrea, is a distinguished pharmacist, clinician, scientist with a passion for translational science. She will talk about initiating personalized phage therapy in Singapore, including phage susceptibility testing. Associate Professor Andrea conducts evidence-based research to improve patient care, focusing on critical care, infectious disease, and antimicrobial resistance. To date, she has authored over 120 publications. Let us extend a warm welcome to Associate Professor Dr. Andrea.
All right, thanks for having me back here since July. Okay, next slide. Okay, this is my overview, how we are going to identify patients, how phage are sourced, and how cocktails are developed. So we have a phage library, we have a phage microbiology platform to work up those phages. I'm just going to walk through our first clinical case, plans for next clinical cases, and anything we have missed for discussion later. So we say we are doing phage therapy for difficult to treat infections, which means unable to remove source, biofilm-related infections. You depleted all your choices in terms of antibiotics, surgeries, and whatever not. And drug resistant, and all drug resistant, big bugs, no drugs, you know. So typically, in a patient journey, all right, we have refractory or resistant infections. If there is a source control is no longer feasible, I can't see my pointer. Okay, never mind. So we need to do, usually we try to optimize everything, TDM to optimize PK/PD parameters to guide the doses, and sometimes we have to go up to beyond the recommended doses by manufacturers. And even, you know, we use antibiotic combination testing where appropriate. So this whole flow or process is also what buys us time for eventually a phage therapy needs to be implemented. Okay? So right from the start, we have the ideal clinicians working with us to quickly acknowledge or identify that this is a patient who may end up having phage therapy. So they will sound the alarm way in advance. This is important because we will then need to keep all the clinical isolates of that patient. So for my lab, when I start doing phage susceptibility testings, I don't like to just use the most recent bacterial isolate. I will want to get hold of all the bacterial isolates in that patient's journey right from the time they had this very difficult to treat infection. So, which is why the initiation has to be pretty early right from the start. They have to recognize that. Okay. So we will archive those bacterial isolates from that patient, and we will use all the TDM, antibiotic combination testing, whatever, to control the case until we find a phage cocktail that we can use to target those bacteria that this patient had in the past few months. So then the other thing is so this is the green part is about our phage infrastructure. We did the characterization. This is the Okay, so this is we continue with the phage characterization with all the isolates that we have collected. We isolate phages. We make sure that from these bacteria, we hunt for them if it cannot be found in our library for the suitable phages. And for phages, newly hunted phages, we have to extract the DNA. We have to send for characterization to make sure all the genomic sequences contains no that's really lytic, no resistant genes, no virulent genes, and so on. Then, okay, so if we found phages, and we found phages that can resuscitate antibiotics, all right, we will let the physician know, and then that is where the whole discussion, a multidisciplinary team will have to be set up to review the case for phage therapy. And it must be satisfied that there is a failure of standard of care treatment, and phage antibiotic salvage treatment has to be offered. So there will be approval for compassionate use, consent, and research consent, because when we start monitoring this patient, you'll be under research. So next slide. So if we zoom down on the patient's perspective, so the patient infected with antibiotic-resistant or difficult-to-treat bacteria, we will continue to get the bacterial isolates from this patient, and then we will screen for phages from the library or from natural environment, work up, and then we will administer to patient when the patient agrees. The multi-D team deems that this patient is eligible to receive phage therapy. So, compassionate use of phage is approved by our Singapore General Hospital Translation Medicine Office under MedBoard. So, the hospital, as an umbrella, stands up to say that we will support phage therapy. We seek consult with HSA, and HSA told us, advised that this falls under the clinical realm of Clinical Practice Act in our country. So, if the hospital is supportive, then you can go ahead for your compassionate use. They are not going to stop you from saving lives if these patients are at their dire straits and there's no other choices left for therapy. So then this is what's going to happen. And when we finish, when we found the phages and we found the phages that can resuscitate antibiotics, that is a point of no return if the patient has decided to go for phage therapy and MDT team has agreed upon to prove the patient for phage therapy. We will go into production. So which means the patient and the doctors has no chance to say that, okay, we withdraw this patient. The only time that we will withdraw this patient is when the patient deteriorates critically and may not wait for phage therapy. So we have a phage therapy referral form for Singapore. So any hospitals can refer to us for phage therapy. So if they have a patient who needs phage therapy, they will have to refer to us. And once we walk through the case, we talk to the patient ourselves. And if the patient is still keen and they do fulfill all the criteria, then they are supposed to transfer that patient from the other hospital to our hospital for treatment, because we are covered by our own Clinical Practice Act, because our whole entire medical board is supportive, whereas they are not. So these are the clinical use of phages. This is not new to all of us here. And the niche role for us in our country or in our hospital are very, these areas: refractory resistant biofilm infections, prosthetic implants infections, LVAD. We have a list of LVAD patients queuing for phage therapy right now. Chronic recurrent infections, we don't have cystic fibrosis, but I do have a HIV stable patients with salmonella infections that is recurring, now episode number 3 or 4, and he has already lost one lobe of lungs, and he can't go for another surgery anymore. So that's another case that we have right now in the queue. So for complicated intra-abdominal infections as well, because those cases are likely escape pathogens with carbapenem resistance, mycobacterium infections, and so the last point, is there a role for decolonization? I believe is yes, but we don't have time to explore that point now. So this is the part that I want to show forth is the lab processes that is involved. I think Jeremy has shown that just now. So phage library, our first source of phages. So this is our library right now, the type of phages, the number of phages we have, the amount of NGS we have performed, and NGS analysis we have done. So we have the most number of Pseudomonas phages, and most of my cases are in Pseudomonas right now, but we do have a Salmonella coming up. This is our phage sequencing analysis workflow. So it's a hybridization kind of protocol, where you need to sequence your bacteria, and in order to sequence your phage and analyze your phage, you need to have the bacteria to minus out. So essentially, what we hope to do is to assemble information, do annotation summary, make sure that there is no VF, virulence genes, AMR genes, integrase genes, and sometimes we use phage AI prediction. We want to form a circle or a linear annotation map. So we know that sequencing is the key to knowing your phage genomic profile. So the quality and safety of phage product, number one, it has to be determined by your genomic analysis first before you can go on to work it up. So the aim of a phage microbiology platform in our case is to look for phages that can infect the intended bacteria or bacteria within the past 6 months that this patient had. So it is to create a phage cocktail that can interact positively to provide sustained bacterial killing, number one. Interact positively to provide sustained bacterial killing when combined with antibiotics if we cannot achieve the first point. Third is to resuscitate or improve the potency of antibiotics. And number four, to attack biofilms. So, like with the rest of the phage lab, we use, this is the way that we can look for phages that can infect bacteria, right? And solid agar method, or it could be a liquid testing method, those we all do, but we also add on a time-Q assay. Because when you look at time-Q assay is needed for us to know the sidality of the phage cocktail and that sidality of a phage plus antibiotic combination. This is what I've just spoke about. So synergistic and bactericidal phage cocktail, synergistic and bactericidal phage and antibiotics combination. So we also do this microtiter MICs of antibiotics with or without phages as well. Okay, let me present a case to walk you through how we present those data for those testing we have done. So we have done this, we have given phage therapy in this case with infected bentol associated with recurrent Pseudomonas aeruginosa bacteremia. So we know, I'm not sure how well versed the audience is in terms of surgical method, but bentol procedure is a very complicated procedure. And when you had an infection out of it, it's like almost a death sentence. And you can't persuade the patient to go back to tolerate another 48 hours of surgery. You're basically telling her that there's no options. So we have this 37-year-old Chinese housewife with congenital heart disease with infected bentol. So pulmonary atresia with VSD since young. So this is her clinical journey. She has been through multiple open heart surgeries. So when she presented to us in 2024, there was already a redo of sternotomy, number 5, bentol procedure, the mechanical valve conduit, the RVPA conduit, and so on and so forth. After the procedure, it's complicated by a pseudomonal acute mediastinitis with anterior mediastinal collection surrounding the aortic root and ascending aorta. So this is the whole journey that she had. And if you see that, after the high-grade bacteremia on February, all right, she constantly kept throwing out infection. There was a time that she went back to OT, and the surgeon just flushed just used ciprofloxacin to flush the heart to try to get rid of the Pseudomonas. But after the procedure, within 2 weeks, she got her bacteremia again. And it's the same Pseudomonas that keep popping out. And sometimes her Pseudomonas infection is pretty asymptomatic. And how we found out that she is actually throwing Pseudomonas bacteremia is by blood surveillance every week. Okay. So if you look at the PET-CT uptake, that is all these months, these PET-CTs are before phage therapy. You can see that the green, red thing up there. Maybe this is better. So you can see that before phage therapy, in the leftmost white column, before phage therapy and after phage therapy, there is a significant difference of the uptake of FDG. And in fact, after post-phage therapy, it's much lighter. So while before we gave her phage, we have to control her from throwing out bacteremia again and again with all kinds of antibiotic in combinations and with TDM. So we do TDM levels for fosfomycin, for cefepime, for piptazole, and we keep trying to adjust her dose, but it is always inadequate to target the levels that we hope for her to reach her site of infections. So this is her journey. So she was a bit depressed. She thought that she had enough of these IV antibiotics. She wanted to go home to spend time with her 6-month-old baby then. So we had to give her OPAT, outpatient antibiotics. So we put piptazole into a pump. We start titrating her dose again. We have her come back every week for us to titrate her doses. And if you notice that actually we have to overdose her for piptazole. It was a 4.5 Q6 run over 6 hours, plus a 4.5 twice a day BD dose. In order to hit at least, the piperacillin concentration to be above 12 mg per liter, and there's enough, assuming that there's 20% tissue penetration, and we are able to achieve the right amount of piperacillin inside the heart area. But we know that with that kind of dose, we are still not able to optimize the tazobactam level. We want to be sure that that tazobactam levels within the heart is close to 4. Again, so we try to add on levofloxacin here to see whether it can synergize with the antibiotic. And we did an e-test to illustrate that. So how we read this e-test is not the way that FICI index or the synergy thingy. All right, what we want to, we ask the question of, if we combine the two e-tests, what is the what's the resultant result? What's the MIC that we can result or reduce to? So, and in fact, that we find that the MIC of levo can be reduced to 0.25, and for piptazole, it can be reduced to 4. So hence, we know that that is a concentration that we should work towards to optimize our levels for. Does it make sense? Okay, so in the end, we control her from not throwing any more bacteremias by giving her piptazole with levofloxacin. So that gives us the reassurance that we don't have to keep chasing after the isolate for our phage therapy. Okay, so then we gave her phage therapy on 23rd of September 2024 last year. While we gave the phage therapy, we also gave her the combination antibiotics as well. So these are the monitoring that we adopt. It's actually following STEM protocol with some modifications. And these are her lab parameters, which is not too bad. She doesn't suffer any side effects as a result of phage therapy. So from the laboratory perspective, we found about 8-9 phages that is possible. And these are the 4 bacteria isolated during her course of journey from January to September that we can isolate. So if you look at the 4 bacteria mixture, the MICs kind of deviate slightly from the rest, but not much. So we look at the sequencing analysis. These are the phages that we found, and they are therapeutic considered because they don't have, and they are lytic. They don't have integrase genes, they don't have AMR genes, they don't have virulence factors. This is the electron microscopy with courtesy from my NTU collaborators who are very excited to give us their phages and even provide a photograph of their phages. So if you look at this, the first isolate that we have from her, that's PA2096. These are the antibiotics, MICs, but when we add phages on top of these antibiotics, you can see that all these phages, some of these phages actually resuscitate the antibiotics or improve the potency of the antibiotics. And if you look at the time Q studies for 2081, which is one of the isolate as well, every single phages cannot effectively kill the bacteria. Same one for the second PA isolate, the third isolate, and the fourth isolate. So if we combine the 4 isolates together, all right, and we start thinking to ourselves, how about we do 2 phage cocktail? This is at best that you can get. So we ask ourselves, how about 3 phage cocktail? Not that great as well, but there is a further cider killing. That means this part here, this part here. So we are looking at this part here to assess which phage cocktail we should be thinking about. So we are looking at the growth. For some of the cocktail, you see that it crashed them to rock bottom, 0 count, but it bounced up still. So we are looking at what is a sustained killing like and for how long before it bounces up. So we look at the 3 phage cocktail on the 4 bacteria mixture, and we found that eventually we choose the PW21KSY1A and P0413. KSY1A is a jumbo PA phage. And these cocktails resuscitate the most number of antibiotics. So once again, we proved that against the mixture of PAs, all right, they actually show heteroresistance resistance to all the antibiotics that we intend to use. But if we combine the antibiotics with the cocktail, the 3 phage cocktail, you can see that they can crash the kill to right 0 without further emergence of resistance. Likewise for this cocktail as well, and this one. So eventually we choose this cocktail because it resuscitates the most number of antibiotics, and we could have more antibiotic options to help with this patient. Okay. So we also did biofilm dispersion assay to make sure that it can disperse biofilm. So in summary, 5 phages were chosen from these 8 infective phages based on the rates of initial bacterial killing, time of bacterial regrowth, final bacterial cell concentrations. These are the lens we use to look at how we choose. We create 2 phage and 3 phage cocktail, but they cannot eradicate bacteria completely. So in the end, we choose 4 phage cocktails from 20 over combinations. In the end, we end up seeing synergistic phage antibiotics interactions consistently for some cocktails with dazocin, levofloxacin, and ceftolozane dazobactam. And we found that there are 2 cocktails out of this. They perform better at biofilm dispersion, and finally, we found that this cocktail chosen, we chose this cocktail because it works well with most selected antibiotics. So there's a lot of thought process behind. And I grow more white hair after this. All right, so we manufacture these phages, and these are the, under the production column, these are the endotoxins level. It's pretty low. And for this patient, we keep asking ourselves, how do we assess this patient? Recurrence of symptoms? She was largely asymptomatic. We only know that she's throwing out bacteremia because we do a blood culture surveillance every week. The inflammatory markers are quite bland to begin with. But we know that there's recurrence of bacteremia. So how we monitor her response is on using PET-CT. So we keep doing serial PET-CT to look at how she's performing. And actually in the first week of December, she has her best Christmas present. She's off PICC line, which is a big deal for her because she was on PICC line for a year. So back to our case, some reflections. Safety, she has no reaction at all. Efficacy, we are not quite sure yet. We have included the role of PET-CT into our determination time point. All right. One of the ways that we look at this is it helps her to defer the need for a high-risk cardiothoracic surgery because she doesn't have to go in for another reopen heart surgery for another 48 hours with a mortality rate of nearly 30 to 40%. So, and it impacts on her quality of life. She doesn't have to come in for IV antibiotics anymore. Her PICC line is out. She's now suppressed by oral levofloxacin. So impact on choice of antibiotics post-phage therapy, we are able to step down to oral antibiotic suppression, potentially taking this off altogether at some point. So the optimal window for referral, for phage referral and treatment is very important because all the phage hunt, antibiotics and phage combination testing, production and all this QA, QC thing takes time. And while it takes time, we also need to find a way to control her to stop throwing out bacteremia because we cannot be catching up with our phage workup. So what next for us? We have an ongoing case right now. It's a refractory PD, peritoneal dialysis, Pseudomonas aeruginosa peritonitis. He has failed kidney transplant, a poor candidate for elective revascularization in view of difficult anatomy, limited options as well. We have a third case following immediately after the second case, which is a LVAD case, so it's all going to happen in February. Thereafter, we have in March, April, we should be having a fourth case which is a post-prostate adenocarcinoma, stable, but he has undergone many surgeries, replacing his PCN because of the left and right VUJ stricture right now. So every 2-3 months, he's going in for a change at OT level to reduce the load of infections. We have a fifth case, like I just mentioned that, Salmonella empyema, stable HIV, virologically suppressed since 2014, but he is undergoing bad episode of Salmonella infection, empyema, and he cannot go through any more surgical decortication already. He has removed 1 lobe of lungs previously from the Salmonella infection, so now he's a poor surgical candidate again, and he cannot tolerate 1 lung ventilation. Okay, that's it. Thank you.
Thank you, Associate Professor Dr. Andrea. Ladies and gentlemen, our last speaker before we break for lunch, Dr. Balint Kinses, is from the Hungarian Center of Excellence for Molecular Medicine. With 20 years of expertise in biochemistry, molecular biology and genomics, Dr. Balint has made significant contributions to the field, including innovative methodologies in genome engineering and microfluidic-based high-throughput screening. Let us warmly welcome Dr. Balint to share his insights.
Thank you. First of all, thanks for the invitation. It's a huge pleasure to be here. And so in the next 20 minutes, I'm going to show you how we plan to support phage therapy by genomic surveillance, and also how nice of a tool is this for people to team up from different countries. There is a movie that I like to show you. I hope it is not going to be too loud, so be careful. Oh, there is no voice at all. Oh, there is. Nice, thank you.
Antimicrobial resistance threatens millions globally, with hospitalized patients particularly vulnerable, as the most resistant bacteria are often found in hospitals, causing difficult to treat infections. Phages, the viruses that kill bacteria, offer new hope in combating these pathogens. However, each type of bacterium requires a specific phage for effective targeting. The potentially hundreds of bacteria types and their unknown distribution in hospitals makes determining the phages needed for treatment very difficult. As a response, we've developed a genomic surveillance pipeline. We start with collecting and genome sequencing bacterial samples from hospitals. Then we add tens of thousands of genome sequences from public databases worldwide. We integrate this with geographic and temporal information in a phylogenetic framework, complemented by high-throughput phage sensitivity profiling. This produces information on the diversity and prevalence of the target pathogen types in the phages that effectively target them, also generating a map detailing the distribution and spread of the target pathogen types. These observations enable the localization of regions and hospitals where the same bacteria types exist, requiring the same bacteriophages. This approach enables the preemptive preparation of region-specific phage collections that target most local infections. Such collections will significantly reduce the time required for administering personalized phage therapy that can become an immediate adjunct treatment for acute drug-resistant infections in hospital settings. Understanding the global geographic distribution of pathogens will be crucial for finding patients who require the same bacteriophage. This will lead to effective study recruitment for validation in clinical trials and for the scale-up and cost-effectiveness of personalized phage therapy.
Thank you. Our first target was A-Baumani, and as Jeremy explained very nicely, the capsule is the primary determinant of phage sensitivity, and so all over the world there are more than 250 capsule types. So this is a huge number, right? And therefore, the estimate was that we may need more than 300 phages to tackle this species. This is a huge number, and this is a huge barrier for phage therapy. So we asked the question, is this really true? How many A-Baumani types causes difficult to treat infections that are carbapenem resistant? Are they really that diverse? What is the geographic distribution of these carbapenem resistant A-Baumani, or CRAB for short, on the different geographic levels like hospitals, cities, countries, or entire geographic world regions? We also asked the question how quickly they spread from one region to the other. How much time do we have to use the phage library? How frequently do we need to upgrade these phage libraries? And also how many phages this library needs to contain. So these were the questions when we started. And so as you saw in the movie, we carried out our local structured surveillance where we collected samples from more than 40 hospitals during the COVID pandemic. So in this period, 10,000 of people died in secondary lung infections in these hospitals in the region. And so we collected and sequenced more than 400 isolates from 5 countries where roughly same number of people lives as in Malaysia. And complemented this set with all the available genome sequences from NCBI. So that was about 15,000 sequences at that time. By now it tripled actually. And then we down-filtered these sequences to a smaller number, removing those that were not carbapenem resistant, or those that are coming from heavily oversampled regions, because we think that these a little bit more than 1,000 sample represents the entire the population, the crab population in that 9-word region, where we had enough sequences to have a good coverage. And then we clustered these genomes. We clustered using 2 typing schemes. One is the MLST typing, that stands for evolutionary distance, basically, and another typing, CPS typing, that represents the chemical composition, let's say, of the capsule. So we use this combined typing scheme, which we call MST-CPS typing, and we ask the question, how many such types cause the infections in that 9-word regions in the last 6 years? So a big surprise was to see that actually a large proportion of the infections are due to just a very small number of MLST-CPS types. So specifically, 50% of the infections in 1-word region are due to 4 types. And for 90%, the same figure is 17 on average. And as you can see here, Southeastern Asia is somewhere in the middle. So not different from other word regions, at least not based on the available genome sequences. Then we pulled this data together, and we were very surprised to see that 75% of the genomes belong to only 31 crab types. Okay, here you can see on this heat map the percentage of those types that are responsible for 90% of the infections in these word regions. Here you can see the identity of these types. The yellow one stands for those that are prevalent in one word region, and the red ones are those that are not only prevalent in one region, but also present in high percentage in multiple word regions on different continents. So these 3 clones, ST2KL2, ST2KL3, and ST2KL9, these 3 together are responsible for 40% of the genomes. This is a very high number. Then we asked, okay, how much time we have with these bugs, how fast the population changes in a given country. For that, we built this phylogenetic tree that contains more than 7,000 crab genomes. And it's a time-scaled tree. So when we zoom into the tree, you can see that the nodes are, we also refer time to the nodes. So we can basically estimate when the transmission lineage was introduced to a given region. So here you can see that the biggest transmission lineage in Eastern Europe, it belongs to ST2KL3, and it was introduced into this region about 20 years ago. And the color coding here stands for cities. So the Romanian cities are blue, the Serbian cities are green, and the Hungarian cities are red. And as you can see, most of the transmissions are happening within the country. So this is good news, that there are relatively fewer international transmissions. The majority of the transmissions are within the country, and therefore the crab population is stable for a while within a country. So we asked the question, how long is it stable? And we came up with a number that for 6 years. So we carried out this analysis that we asked, was the chance that 2 isolates that diverged just before 6 years, or not later than 6 years ago, was the chance that 2 isolates are in the same country or in different countries? And we saw that within a time frame of 6 years, it is 10 times more likely that the two isolates are within the same country. But over a longer time scale, after 6 years, this difference disappears because of international transmissions. We see a similar pattern on the level of cities, just on a shorter time scale. So within 2 years, isolate pairs that had an common ancestor not longer than 2 years ago, the chance that the two isolates are in the same city is 6-fold higher. But then this disappears. And this disappears mainly because there are new strains coming into those cities, and it is 4 times more likely that the new strains are coming from capitals and major medical centers and not from smaller cities. So if we put this together, then we can say that what dominates the infections today in the capital and in these major medical centers, then those are going to be the strains or the types that are going to be all over the country in the coming 6 years. So then we asked, okay, how many phages do we need to tackle those types that cause 90% of the infections in our region? And so in our region in southeastern Europe, we identified 11 crab types that cause 90% of the infections. And we used those in the phage hunt from sewage. And we came up all together with 15 phages. And here you can see the host range of that 15 phages on 96 isolates. And the isolates are grouped according to their MLST CPS types. For example, here you can see ST2KS3, the most prevalent one in our region. It's quite homogeneous in terms of phage sensitivity, independently of which city and country they are coming from in our region. We weren't that lucky with the second most prevalent type, which shows a much bigger heterogeneity in terms of phage sensitivities. So then we started to put these phages together to cocktails, not only to broaden the host specificity, but also to mitigate phage resistance. So the principle is the same what Jeremy just showed you, that some phages target the wild type, the capsulated one, and we combine those with other phages that has a different receptor, and they are effective against the decapsulated version. And so in this way, we can make phage resistance, let's say, go away for a longer time scale, even though we don't know what's the clinically relevant time we need to follow, but we followed it for 72 hours. Then we used these cocktails in vivo infection models in 2 types. One is the Galleria melonelli infection model, and the other one was mice intraperitoneally. And as you can see, the animals were saved in both cases. In case of the mice infection model, all the animals survived. While in the absence of the phages, they all died 1 day post-infection. Then we repeated the same procedure for 9 MLST CPS types in our region. And out of these, 4 are actually global clones. So they are prevalent not only in our region, but also globally. So we hope that these cocktails can be used somewhere else as well. And then we validated the whole model by concluding another genomic surveillance study. So we collected 157 new isolates 2 years after the conclusion of the initial surveillance. And we sequenced those, and we saw that 90% of them belong to the MLST CPS types that we saw in the region before. So that validates the epidemiological model. And we could offer 8 cocktails against these 8 types out of the 9. And then we tested the individual cocktails against the corresponding isolates, and we saw total suppression in 81% of the cases and partial suppression due to phage resistance in 19% of the cases. However, I also like to add that those isolates that start to grow in the presence of these cocktails have large fitness costs. They become sensitive to antibiotics sometimes, and they also become virulent in our infection models. Okay, so finally, I pulled together some data, what is available for Southeastern Asia. There are a little bit more, or roughly about 1,000 genomes available, publicly available in NCBI. And as you can see here, the majority are CRAB, carbapenem resistant. Those are the green ones. Unfortunately, there is a drop in more recent years in the number of sequences. Maybe the COVID had an effect. And here on the right side, you can see the so-called rare fraction curves that shows how many samples you need to collect basically not to discover new MLST CPS types. So that just shows where it saturates that your surveillance is good enough, has a good coverage. So Thailand is having 250 isolates, CRAB isolates, corresponding to 30 types, and it looks as if that they already covered most of them. Here you can see, it's not possible to read, but that's China. China already produced 1,500 sequences, and it saturates roughly about 40 MLST CPS types. And here is Malaysia. You already produced a little bit less than 100 sequences, CRAB sequences, and they belong to altogether 11 MLST CPS types. So here you can see the identity of the same heat map for Asian countries, where we saw enough sequences. So the countries are clustered according to their CRAB population similarity. And as you can see, Malaysia clusters nicely together with Singapore and Thailand. And I think the CRAB population is also very similar in Vietnam, in Myanmar, just they haven't produced enough sequences to form one cluster. But as you can see here, the clones are very similar that are present in these countries. So these are also global clones. I indicated those that we also have in the region. So there are 3 ST2KL2, ST2KL3, and ST2KL32, for which we already have a cocktail. Yeah, and that's just testing one of the cocktails on a larger set of isolates. So we are continuously developing further our cocktails to work on a lower MOI and also to contain fewer phages. And so with one of the collaborators, we could measure on a truly global collection of ST2KL3 isolates one of our cocktails, and one of the isolates is from Malaysia. It's actually now with the new tool, it is predicted not KL3 but KL150, but it's more or less the same capsule. And the cocktail is effective, and there was another isolate from Vietnam where we also see about 90% suppression. So this is promising, and it just shows that one phage cocktail or phage can be used on multiple, at least in vitro, show suppression on multiple continents, which is just promising and shows that there is a chance that we can scale up. And if we manage to produce a phage batch, we may be able to use it for multiple patients and share it globally, just as John explained. That would help a lot on the financing of phage therapy. Okay, so, oh yeah, one more thing, forgot it. So here with these arrows, you can see the Malaysian transmission lineages in the tree, in the phylogenetic tree. And one of the lineage, the ST2KS3 lineage, is very close actually to the Hungarian one. So we just figured out that you guys received this ST2KR clone roughly at the same time as we did in Eastern Europe about 20 years ago. And they are really closely related. So it's very interesting to see. And it also helps, so this type of analysis helps, you know, which countries you guys need to team up, in which countries you share the clones and come up with a solution together. Okay, so in general, genomic surveillance helps you to find hospitals, cities, or even regions where the same phage may be a benefit. It also helps you to define the market size of the phage, so you can prioritize which phage to put more energy into. Also, it helps to recruit patients for clinical trials. And as explained before, it may help to make a broken business model viable by just scaling it up. So finally, I'd like to acknowledge my group who carried out the majority of the work that I showed you, and all the collaborators who kindly provided us bacterial isolates. And thank you for your attention. I'm happy to take questions.
Any question from the audience? Yes, Dr. Toby.
So on average, we have 3 phages in the cocktails, but we are trying to decrease this number to 1 or 2, we'll see. Just to be able to combine more phages at the end to have broader coverage. Yes.
Are you able to distinguish the MLST type to the KL type in terms of phage infectivity?
Yes. So we do see that the capsule type is the major determinant, albeit the ST type has some effect on phage sensitivity, albeit it's much weaker. So you can find phages that work across MLST types, but not all of them. So the genetic background has some effect, but not on all phages. Some are stronger in this respect. Yeah. John?
Yes.
Just wanted to ask about the stability of the capsule type. I asked you before, I think, in a taxi or something, whether you thought prophage mediated changes to sugar residues were important, and if your capsule types are really stable, it implies they're not, unlike for other reasons.
Yeah, so, okay, my slides are gone. But yeah, so the change over time, we haven't calculated properly, like that's one of the next projects. We don't know what's the selection pressure, but we do see that capsule type frequently changes. Oh, that's great, thanks. Oops. Okay, so it's difficult to see from here. I'm just going to explain it. So we see that capsule types frequently change at the same time of transmission from one place to another. So we see that frequently, yes. We don't know if it is related to, you know, what's the difference? Maybe selection pressures are different in different regions. And so we would like to, it's just a feeling, we would like to properly analyze this, if this is really the truth. So within one region we see rarely the capsule change, but it does happen, it does happen.
Any more questions? No more. Thank you so much, Dr. Balin, for sharing with us. Ladies and gentlemen, now it is time for our lunch break and also prayer break. Our next session will begin at 2 p.m. So you may come back to this hall 5 minutes earlier. You may proceed to the same hall for your lunch. Thank you, everyone.
Good afternoon, Salam Malaysia Madani. Welcome back, ladies and gentlemen, to the Malaysian Stakeholders Symposium for Phage Therapy. I see we've all had a satisfying lunch thanks to the organizing committee. So I think without further ado, we'll just kick on and continue with our next speaker. Our next speaker is Yang Berusaha Dr. Frank Smreka. He is the founder and CEO of Jafral, the world's largest CDMO specializing in phage-based product manufacturing. Under Dr. Smreka's leadership, Jafral has built an impressive track record producing biomolecules like pDNA, mRNA, OMVs and recombinant proteins, as well as providing sterile fill and finish services. Dr. Smreka has played a pivotal role in developing products that have advanced to clinical trials globally, including in the US, European Union, and Australia. His expertise ensures compliance with stringent regulatory standards from bodies such as the FDA, EMA, and TGA. Let's give a warm welcome to Yang Berusaha Dr. Frank Smreka to share with us on GMP phage manufacturing.
Because it's that's better. Because it's after the lunch. If somebody is going to fall asleep, it will be because of the lunch, not that I'm boring with the presentation. So hopefully not too much. Okay. So thanks for nice introduction. It was said already a few things. I'll just on the few slides say what we are doing. So we are what's called contract manufacturing organization for biologics. We are located in Slovenia in Europe. We are actually really the world's biggest phage manufacturer, and production of GMP and non-GMP products. Majority of our activities, like around 80% of our activities, are production of human therapeutics. Then we have also something for food industry, cosmetics, veterinary, and agriculture, and for each of this we have also appropriate certification for that. So yeah, if it's not the closest to Malaysia, but if you're ever around, if you're visiting maybe Berlin, drop around. We'll have a coffee or stuff. So we have 2 GMP facilities for production of biologics. But if the phages are natural biologics, then natural phages, then they fall into biologic field. If the phages are GMO, then they fall into ATMP field. Then there's also developing, as mentioned already today, phages for other fields, which is food supplements, cosmetics, agriculture. Of course, in those cases, the path to the commercial is much faster. So in that cases, we do have commercial products where for human therapeutics, what we produce now is phase 1, phase 2 material. In the last year, actually, we also increased the facility to 200-liter scale, which enables also then the phase 3 and commercial production, having then also by safety level 1 and 2 in non-GMP. So far, we produced more than 50 phage banks and bacterial banks, over 200 GMP batches and 12 drug products, which is the most that somebody produced for the clinical trials. Most of the trials are in US, then also some in Europe and Australia. And all of this was then also reviewed by different regulatory authorities. So we had to submit also, it's not only the product, but also all the documentation that is needed to go into clinical trials. And being in the manufacturing, lead in the manufacturing field, we were then also invited by EMEA, FDA, now EDQM for preparation of legislation in the recent 2023. In Europe, there were some changes in the EU phage regulations, so also for magistrate products, we were also part of the preparation of that documentation. The track record of very differently produced phages, we work with typical suspect, the escape pathogens, but also with number of other bacterial phages, and nowadays where also new types of the application of phages come around. It's not only the maybe aerobic bacteria, but also as a microbiome and stuff like that, also anaerobic bacteria, and then also the phages. So going to the to the manufacturing side. So how everything starts is, as you all know, so you start with bacterial infection, then is the phage selection, isolation, or phage design if the phages are GMO. So then what we are doing is small-scale process development. And maybe also to separate here, so if we are doing the compassionate use or just producing phages for one patient, in that case, the production process is quite straightforward. We have the platform process for production with all the quality parameters, and we're using that one. If the product goes into development for the clinical production, in that case, the final material has to be able to be scalable to high quality and also high efficiency. So for that one, what we need to do is development of the process. So what are the best steps to purify material? Then this material, once the development or the process is developed, then also it's called small-scale production. This material, which is representative, is then used for preclinical studies, for animal studies, different in vitro testing. What we always saying is to put the material as early as possible also in the stability program, just to figure it out whether the phages are stable, because at the end of the day, even if you have the best phage or the best product, biologics, if it's not stable, it doesn't help you. So this is our suggestion to do it as early as possible. Then what we are doing is the scale up to different scales. So if we are starting the development to 1 liter scale, then we're going to 10 liter, 100 liter, and 200 liter scale production. Again, stability testing, and then if it's for human therapeutics, it's GMP products production, and if it's not, it's then non-GMP production. So the same part is not, one is manufacturing part, but the same part is also for analytics. So for whatever we are doing, of course, we have to analyze the stuff all the time. We have 80% or 90% of analytical methods are in-house. We are outsourcing some stuff as sterility testing, but nevertheless, as the phages they have different properties, we have the development of the analytics in-house, and either it's just a potency that we optimized, or you're doing the PCR or the qPCR analysis. That's the first step. Then it's method qualification, validation, then for routine usage, and that has to be within the different ranges. So at the end of the day, depending of the product and depending of the application, special application, then there are different quality parameters that you want to know if that your product is within these ranges. So they're typical like potency, morphology, identity sequencing, and then how much you remove impurities really depends of application. If there is the product is used as IV, of course you have to remove not only the endotoxins, but also the host cell proteins or host cell DNA to the high level degrees, and then also sterility needs to be tested. If the phage application is maybe only on topical application or even not of humans, then we try to adjust the process. That is less costly, but also the quality parameters are then different, because maybe you don't need to have as pure as possible, so it doesn't make sense to have really highly purified material. Then when there is a product, you have the phage, you have the right quality. The important part is stability and formulation development. So as said, the product has to be stable, and when we are doing the stability testing, we say it has to be the most boring part, which if it's not boring, that means that something is happening and we don't want to do that one. And the phages are, because they are biologics, they have different specialties. So I don't know, you have for Pseudomonas phage, one phage will like magnesium, the other one won't like magnesium, but if you're doing the cocktail, you have to figure out the conditions that work for all the phages. Typically, at least for the cell banks, we are storing at -80, and the cell banks are usually, of course, non-purified material. where then for the final product, when there's purified material, mostly it's 2 to 8 degrees for long-term storage. But then as for GMP production, it has to be done also then at accelerated conditions, that is 25 degrees, and then also 40 degrees. That just, it's per regulation that you have to do. For Malaysia, definitely it would be interesting also to do the 25 or 37, because at one point, yes, you have the refrigerated conditions, but for short period of time, it will be at the room temperature. So you want to be sure that the phages are stable also at that temperature. The final products, these are just examples. So majority of our products are in liquid. Also for the GMP production at the moment, we have only liquid products, which is then, as said, applied as topical, as IV, as nebulized material. Then for other applications, also for the food industry or cosmetics, we have then also spray drying lyophilized material. We are planning to implement that also in GMP world, also the lyophilization, because at the end of the day, if hopefully phage therapy will be able to help patients as we all wish for, hopefully it will be in the capsule or something like that, so it will be easily applied. And this is also one important part, because sometimes it's big focus on how the phages does work, but we forgot that if they are the best phages, but they are not applied correctly, or they don't survive until where the bacteria is, it doesn't help. So you can lose phages there. So this final part, formulation and fill finish, it's also important. Then just a couple of 2 slides. I know it's a busy slide, but nevertheless, I think it's works here. Yeah. So maybe just to easily or easier explain the GMP activities or non-GMP. So technically the GMP is just specific regulation that for all biologic you have to follow in order to have at the end of the day the license that product was done according to GMP. For us, we can do both, so it's not preference, but from my perspective, why GMP was implemented is just that you have some security that everything complies with some safety measures. And so also the manufacturing scheme is different. So for the GMP part, it's here. So it's typical also what is used also for other viruses. It's a 2-tier system. So when we say we first have the RCB banks or our phage banks, then it's production of the master cell bank and master phage bank, or sometimes it's called master phage seed. In the early stages of the clinical trials, it doesn't need to be done also the working cell bank and working phage bank. That can be, if the product is successful, that can be produced later on, but initially it's not required. And of course, all of this has to be done also in stability, because you want to be sure that you are not losing the banks, because at the end of the day, so hopefully the product is successful and you have the product on the market, it's commercial, you always have to start the production with these cell banks or the phage banks. So it's important stability. Then in parallel, when this is done, it's the process development, analytical development, which I already mentioned. Then it's production of drug substance, engineering runs. Engineering runs, that's just that you test the process in the right scale and also in GMP environment. The other point is also that because once you put the drug on the market, you have to know the expiry date, so how long the stability is, the product is stable, and has to be a representative material. So in this case, these engineering runs are used for that one. Once these are done, then the clinical trial material, DS and DP, and of course, if there is a phage cocktail with more phages, you have to do more of them DSs in parallel, then you combine in the DP. And in most of our cocktails that we produced are mixed into DP. We had also the cases where we produce the DSs and the DP or the mixture is done at the site of the patient. Where if we see then the non-GMP part, it's much less much less steps needs to be done to the final product. But actually the most part, the work or the cost for doing the GMP is actually what needs to be done to assure that one. So I won't go into detail, but maybe just the main part. So with the GMP certificate, you're putting, as a manufacturer, there's assurance that the product is produced according to some standards, which means that, I don't know, the clean rooms, the premise clean rooms have to be validated. So IKO, QPK, so in that every year it's revalidation. Also then the whole equipment has to be validated, and why, for example, this is important. I don't know, if there's like, sometimes explain, there's like autoclave, some sterilization process. If you don't revalidate every year, you maybe something would fail, and then the product, if that is the critical part, wouldn't be sterile at the end. So all the equipment is validated. Then there's like a quality system, documentation, change control. Just for that one, we have, I think, like 400 SOPs to monitor everything, and then every 2 to 3 years you have to renew that one or check if something is changed. Then the QC methods, the quality of the QC method, they need to be qualified, and then also we need the positive controls, negative controls, stability programs. Then biopharmaceutical quality of raw materials, and again, that is also, we are sometimes joking that we maybe did go too far, but for example, if we are using the sodium chloride for the production or whatever media, even though if it's purchased from the Merck or some supplier, biopharma, then we have to go back and send for additional testing that they confirm that it's really sodium chloride. Sometimes it's fun, but yeah, most of the time it's just to double check that everything is as it should be. So, and then also the personnel has to be trained, and the production has to be tested. So all of this, once somebody asks us why it takes so long to produce GMP material and so expensive, all the things are done here. As said, from our perspective as a company, we say, yeah, we can do GMP or non-GMP, but if GMP, this needs to be done. If non-GMP, just everybody that use that one has to be aware what the product delivers in the same time. So that's just in brief. So, and that's all from my side. That was just short introduction of manufacturing in GMP. So, if any question, but if not, thank you for the opportunity.
Thank you, Yang Berusaha Dr. Smreka. I've always found it very fascinating to be taken through the whole process of translation and transforming a vision into the end product. So that was very interesting. So before we move on, just a quick note, just for your information, our symposium today is actually also being streamed online, live on YouTube, and we've had about 100 participants tuning in from all over the world since morning. And also there's a link on the YouTube page for any of you to ask questions to any of our speakers. And if you ask a question through the link, we'll pass it on to the speakers and get back to you. Okay, so for the next talk, we'll be switching gears a bit to the regulatory aspects of phage therapy. We are honored to have with us today Yang Berusaha Dr. Toby Nagel, founder and president of Phages for Global Health. Phages for Global Health is a non-profit organization dedicated to advancing the use of phages as antibiotic alternatives in Africa and Asia, regions most heavily impacted by antimicrobial resistance. Dr. Nagel brings over 15 years of pharmaceutical industry experience, having co-developed drugs tested in more than 80 clinical trials. As a Fulbright Global Scholar, she has been gathering insights on phage-based drug regulations in the US, Europe, and Australia, and sharing her findings with leaders worldwide. Dr. Nagel has also contributed as an NIH ad hoc reviewer for bacteriophage therapy and serves on the advisory board for Phage Pro, a company focused on cholera phages. Please welcome Yang Berusaha Dr. Toby Nagel to the stage to help us explore the overview of phage regulatory systems in different countries.
Thank you very much. And thanks to the organizers for doing such a great job, and for all the speakers for being willing to come, and for hosting us here at NIH. I'm really enjoying all of this. So let's see if I can. This is why we're here, right? Because by 2050, 10 million people are going to die each year due to AMR infections. But what's often lost in this figure is the fact that 90% of those deaths are going to occur in Africa and Asia. Africa will have the highest per capita death toll, as shown by the, well, it's not quite working, as shown by the color. And then Asia will have the highest actual number of deaths. And so at the time we began our work at Phages for Global Health back in around 2016, most of the phage experts, and perhaps it's still true, are in North America and Europe, as well as down under in Australia. And so simply put, the mission of Phages for Global Health was to bridge that gap, is to help empower phage product development in Africa and Asia. And so we do that through 4 different pillars of our work. We run laboratory training workshops. We collaborate with different groups around the world to develop products for application in Africa and Asia. We are helping to set up guidelines, international guidelines for phage banks. And what I'm going to talk about today is the regulatory component, just that piece of it. And so, as was mentioned, I'm doing this as part of a Fulbright Global Scholar project where I'm compiling information from these high-income countries in different regions about how phages are currently being regulated. And that information comes from 3 different types of information. Formal guidelines, of which there are not that many, I'll talk about those. Informal guidance, where, say, people from the FDA have presented at conferences or have personal communications. I'm often emailing them to ask their specific recommendations or questions. And then I've also interviewed many companies that have gone through regulatory processes in different countries to ask what's their actual experience, what's the data that was actually required of them. And so it's pulling together all of that information, and then with this Fulbright, I'm sharing that with lower and middle income countries, specifically these 3, Kenya, Malaysia, and Uganda, 2 months each. So I was actually here in Malaysia for 2 months, hosted by Drs. Hira and Siva at AIMST University back in October and November. And I'm also, I will say, starting to work with South Africa because their government, like Malaysia, has decided that they'd like to sort of have a national phage plan or infrastructure in place. And so I will actually be going there after this to start working with their government as well. But the overall goal here is to say, let's benefit from how things are being done overseas, but see what might be relevant, make informed decisions for what might be more relevant in Africa or in Asia. The reason being that your needs are different and your infrastructures are different, and frankly, you will have an almost 10-fold higher death toll from AMR than my country will have. So your urgency will also be different than ours. And so let me start, if you take home no other information, this is a key take-home message. Clinical trials do not require formal phage guidelines. And the reason I say that is that at least 35 clinical trials have been initiated in 16 countries, and the vast majority of those countries do not have formal guidelines. And I totally agree that investors usually say, we don't know the regulatory system, so it could be risky to invest in phage product development. I think that's a myth because we've got 35 clinical trials going in 16 countries that hasn't held them back. And let me point out that not only do most countries not have formal regulatory guidelines, my country has no formal regulatory guidelines, but I think more clinical trials are going on or have begun in the US than in any other country. So you don't necessarily need to have specific guidelines on how phages would be regulated. What usually happens is, and same in my country, is they figure out how phages fit into the existing regulatory systems. And typically that's as biological products, as Frank mentioned in his talk. So if you are interested in seeing where those clinical trials are happening, what they're targeting, and how they're dosing those phages, you can go to clinicaltrials.gov that I think maybe John mentioned. Most clinical trials around the world for any kind of drug are registered there. Not quite all of them, but most, and you can get a lot of information out of that website. So let's start with the US FDA. again, no formal guidelines, but based on informal guidance and previous decisions, here's the data or the information they want specifically on the phage product typically. They say that you can have a preset cocktail, or they have given approval to test a phage bank, a collection of phages, as your product, where you would pick the specific phages for a given patient in a more personalized approach. Then they typically want to see data in 6 categories. One, strictly lytic, that's already been talked about by a number of the previous speakers. They want to see the genome data not only for the phage, but for the bacteria that you use to produce the phage, to make sure it's lacking not only the lysogenic genes, but toxins, AMR, virulence genes, that sort of thing. Then they also, of course, want some data showing that your phage works against the type of bacteria you want to target. And as has been talked about, they want to see purity. So you're using bacteria to produce those phages, then you have to get rid of many of those contaminants, endotoxin and others. And so the purification processes that others have talked about, and then there might be some chemicals involved in that you also need to get rid of. So that purification really becomes a big focus in what FDA wants to see. Stability, Frank just talked a fair bit about that, and I will highlight this because there was a big clinical trial, Phagoburn, in Europe a few years back that failed, unfortunately, because they didn't do the proper stability testing, really. And so they ended up giving much lower doses of the phage to the patients than they had planned, and they only sort of figured that out after the fact. And then this is the one piece of information, transduction data, generalized transduction rates, that not all regulators require, but the US FDA does want to see that. Some do, some don't. And often what they're allowing is that you have some kind of biomarker for a bacterial gene, and you see the rate at which that's transferred in your experimental system. And often that's enough data for them. So let me also say that while GMP manufacturing is required for clinical trials by the US FDA, they officially have what they call a sliding scale for GMP, meaning your GMP requirements might not be as strict for, say, phase 1 clinical trials as for phase 3 clinical trials. And so they recognize that you're getting more of your manufacturing systems nailed down as you go through larger manufacturing processes. So, and I will point you to these references from the FDA for where a lot of this information comes from. So that was for the product itself. Then the FDA also wants to see certain things for the protocol and the processes. You don't have to do this, but they prefer that you pre-specify as much as possible. How are you going to deliver the phage? What's the pathogen and what's the infection site? And again, if you're going in with a phage collection, a phage bank as your product, maybe you can't fully identify which specific pathogen or infection site you're going to go after. But as much as possible, they'd like to see that focused. Then you need an appropriate screening of your patients, and I think this is really important too, both inclusion and exclusion criteria. Even not in the phage field, but another drug development I've done in the past, I've seen clinical trials fail because they didn't have appropriate inclusion or exclusion criteria. So that can make or break a clinical trial. Then, and I think this is a key part of the STAMP protocol in Australia, you want investigators who are qualified to perform the studies and the procedures, and really make sure you've got the right people doing that you define and monitor for adverse events, of course, and that you have reasons to stop treating an individual patient or to stop the entire clinical trial, that you predefine what those issues might be. And then, of course, the ethics approvals will vary by your country and your institution and your national laws, but in general, you need some kind of institutional approval, informed consent, and also keeping patient confidentiality. Now, this is an interesting thing that the US FDA has pointed out. They have explicitly said that they're not convinced that studies in healthy patients would provide relevant safety data. Because if you don't have the bacteria in the patient, then you don't get the amplification of the phage. So then you're not really testing what might be a toxic event. Not that we're expecting any, but what might be a toxic event. But they do say, well, maybe those studies in colonized patients might give you some information on showing that, for example, the phage doesn't alter your microbiome, and it has that benefit as compared with, say, antibiotics that might destroy your microbiome. So that's an interesting difference of phages than, say, of conventional drugs. And again, I'm pointing back to the same reference for this from the FDA. So now let me go over and talk about the European Medicines Agency, EMA, which was established to harmonize but not replace national regulatory bodies across Europe. And so they have a process of establishing their formal guidelines where they start with a concept paper that they put out for general public consultation and feedback for about 4 months. Then they review that and ultimately publish their formal guidelines. So they first did that for phages for veterinary applications. They put out the concept paper in early 2023, and then they published their final guidelines in October, late 2023. Their human applications of phages, they put out the concept paper that closed early of last year, and it's now under review. So anytime soon, we expect to see formal guidelines published for the human applications. It's anticipated that they will largely follow the veterinary applications. So let me talk about the veterinary applications. And keep in mind that under the EMA, each country still has its own jurisdiction to say, yes, we agree with the AMA, or we want to have slight differences. So this is, in a way, an overall guideline, but might differ in different countries. So for the veterinary guidelines, they require for each phage the same data that the US FDA requires, essentially those same 6 data points: lytic, genome evaluation of both actually the phage and the bacteria, potency or host range data, purity, stability, and they do require transduction data. They suggest you might also want to include, does your phage or your phage cocktail have activity on biofilms? Is it antagonistic or synergistic with antibiotics? Do you have any information on how frequently resistance occurs or what those mechanisms are, so that maybe you could even add additional phages to your cocktail to overcome those resistance, or have a second line cocktail ready to go if resistance were to come up? And then again, for each bacterial host, as I said, they want to see genomic evaluation for things like the toxins, the AMR genes, prophages. They would prefer no prophages as much as possible if you can find a host strain to make your phages that doesn't have prophages, or any other, I love this, any other genetic elements that could be detrimental, sort of a sweeping statement. But they also have this sweeping statement: if freedom from these elements is not possible, it should be justified that this has no negative effects. So there's some flexibility and recognizing the variability of phages and host. So then there's some overarching themes, if you read all the details of the EMA guidelines, that I find are really helpful to emphasize. One is that they explicitly say phage-host interactions are dynamic, so regulation should be adaptable. And here's some exact quotes from the guidelines. A flexible composition of phage products is expected to be the usual situations, so your cocktail might vary in terms of which phages are in it. Manufacturers may pick monophage components from those included in the approved dossier to match the geographical distribution and phage resistance patterns of targeted bacteria in different countries or for an individual. And different compositions of the parental product can be marketed at the same time or at different times in the same or different countries to address different epidemiological needs. So I think this is addressing all of the things that our previous speakers has talked about. And this is where regulation for phages is recognizing you need to be more flexible for phages than you do for a standard small molecule drug, for example. Then another thing is, because you've got this swapping in and out of phages as an allowed part of the process, they really want to emphasize that you've got robust manufacturing to ensure that the quality of the product will be there at the end of the day, even if you're swapping in different phages. And so they distinguish between the upstream part of the manufacturing, so the amplification of the phages in a particular bacteria might have to be changed if you need a new bacterial host, for example. But the hope is that the downstream process is relatively robust for purification and formulation so that you've got still a pure product that's always coming out. And then finally, the EMA guidelines specifically say that these can be natural, adapted in vitro, or genetically modified. Not a problem under EMA guidelines to have genetically modified phages. Not all countries agree with that, but the EMA guidelines say that genetically modified phages are fine. I will say genetically modified phages are fine under the FDA process as well. And then there's some other parts of the toxicology requirements for phages that are different than for conventional drugs that they call out in these EMA guidelines. First of all, they say safety concern is relatively low because phages don't propagate in human cells. Animals and humans are naturally exposed to high amounts of phages all the time anyway. They're already part of our microbiomes. And studies suggest that phage lysis will not release higher endotoxin than antibiotics already might. So sometimes, you know, if you've got a huge killing of a lot of bacteria, you could have a huge release of endotoxins that the body might have some problems with, right? Phages are considered that it's not going to be any worse than what antibiotics are already doing. So genotoxicity studies are when you take your drug and see if your DNA is mutated by your drug. That's required for all standard drugs that are made, but it's not required for phages under these new guidelines. Again, recognizing that these are a different and very safe drug. Also, you can do safety studies or toxicology studies at low doses. So the way these are usually done when you're developing a regular drug is that you give very high doses, higher than what you would usually give to the patient, to an animal. And then you see what are the side effects in that animal by looking at all of their organisms after you harvest the organs, and look at any damage to the pancreas, to the liver, to whatever. They're saying you don't have to do that for phages because we inherently consider them to be so safe. You can do your safety studies at 1x dose. You don't have to do it at really high doses. And you don't have to do postmortem examination of all of those organs. So again, I hope the message is coming across. These regulators are really considering phages to be inherently safe. This I also found interesting. You can do your safety studies with some phages and then extrapolate those results to other phages. Again, this allows for the swapping in, saying that you don't have to do your safety studies with every single phage that would be in your cocktail. They do, again, come back to the robust manufacturing process so that you really get purity. None of the bacterial bits, hopefully, or low quantities of the bacterial bits left after the manufacturing, or any chemical residue. And then finally, they do say should be addressed. Immunogenicity should be addressed. But there's already some data in the literature showing how the body reacts to phages. And that literature data is often considered enough. You don't have to go back and generate it for your own specific phages. So the take home here is it's remarkably flexible. I was surprised when I actually read these guidelines when they came out. And so that's a standard clinical mindset for a large clinical trial for GMP phages. Then in 2018, what's often referred to as the magistral phage system was first established in Belgium. And this is important to speak about because Australia and other countries are using this. And so their regulatory agency approved this monograph that said a pharmacist, like Andrea in Singapore, could provide natural phages, not genetically engineered phages, but natural phages as the active pharmaceutical ingredient, or the API, which is usually the drug substance, with 3 conditions. One, that you have a prescription from a physician for a pre-named patient, and the product must be approved through an independent government lab that they call Cienzano in Belgium. And what Cienzano does is they especially look at what they call the genomic passport, where they want to, again, make sure that your phage is lacking lysogeny genes, AMR genes, virulence genes, that kind of thing. But they also look at lot purity of your phage production, potency, and stability. And then, but keep in mind that this is not GMP. because this is phage produced for an individual patient. It wouldn't make sense to do a huge GMP batch manufacturing for every individual patient. That would be financially impossible. So Belgium is the first country to allow phages that are not GMP as a standard treatment for patients. They also do not require transduction data. Now, these Belgian magistral phages that have gone through and been approved through CNSANO have been exported to 12 other countries with approval by those in-country regulators. This magistral system was actually incorporated into the European Pharmacopoeia in May of 2024. So what that is a rule that says drugs made in Europe, if they conform to this monograph, they are acceptable to use across all of Europe, if the in-country regulator agrees with that, okay? And then just a few months ago in November, Portugal became the next country to officially approve this magistral, non-GMP, personalized phage product development system. And they specifically refer to the Belgian monograph in their guidelines. So let me talk again about the STAMP protocol that's been mentioned before, standing for Standardized Treatment and Monitoring Protocol. This allows for personalized phage therapy by enrolling in this clinical trial in Australia. Officially, it comes, my understanding is it comes under the Special Access Scheme, which is what your equivalent of the compassionate use system. It's a clinical trial, but there is no placebo group in that all patients receive phages. The whole point of this clinical trial is to get the phages into the patients. It does require appropriate medical facilities and staff expertise. So like the FDA, they require that you've got the knowledgeable people and the appropriate resources in place. The phage product can be as per the Belgian magistral system or GMP, so you can use either. Treatment is still typically for 14 days, or is it less now? 14-ish days. Typically IV, though there can be variations depending on the patient. And then I think what is unique about this clinical trial is really the monitoring piece as well, that specific time points and monitoring outcomes need to be recorded at different times, essentially for the first month. And that's great because it gives you information to inform eventually how your country is going, what's going to be the best practice for phage therapy in their country. So this is something that we've talked about as a possibility for use in Malaysia. It both is a clinical trial, but it allows for compassionate use. So it's a little bit of an in-between that might be a good first step. So let me just take the last few minutes to talk about non-human applications of phages that a few others have referenced. Poultry, livestock, and agriculture, in addition to food and crops, have been where those have been officially marketed. And I underline the officially marketed because for phage applications in food and crops in many countries, you don't actually need official approval to sell the drug, but you do have to go through an official marketing process that usually involves a validation, if you will, by the regulators, okay? And these are the two leading companies in the world, Intralytics and Microbial, that have a whole panel of phage products that they sell in a number of different countries. And then that's for post-harvest or slaughter or on food production services. And then you've also got some that are being used for pre-harvest. Omnalytics is perhaps the oldest company around. They're in the US, I think they were more recently taken over by PhageLux, which was originally out of China. And then A&P Infotech, I think, is the newest company on the market. And their phages are for vineyards in my home state in California, where you want to keep your vineyards from getting bacterial pathogens. And so it's a sort of preventative almost market. And so I want to talk for a minute about Intralytics and their experience of getting official marketing quote approval for bringing their products to market. their first product was against Listeria in 2006, and they have since then approved products for E. coli, Salmonella, Campi, and Shigella. And they have used 4 different pathways to get regulatory validation. The first 3 pathways come under the US Food and Drug Administration, and those are either called a food additive, a food contact substance, or generally recognized as safe. Their product can get any of those categories, and it depends, I think, on how the product is applied, at what stage in the food processing process. And they've actually written this paper that I find is really helpful about the different ways that food products have been approved. And then the 4th one comes under the US Environmental Protection Agency, where they call it a microbial pesticide. So you're not using your sort of standard chemical pesticides, but instead microbial pesticides. So each country might have their own versions of these different validation instruments for food decontamination products. Then let's talk again about officially marketed phage products for poultry, livestock, and aquaculture. And there are 3 leading companies that I'm aware of worldwide. One is Proteon Pharmaceuticals. They're out of Poland, but I believe they sell their products in a number of different countries, either for poultry or fish. And for poultry, they have different products that target either, for example, E. coli or Salmonella, given as a feed additive. And for fish, they have a product that targets at least these 2 different bacteria, Aeromonas and Pseudomonas. Here, it can either be sprayed on the feed or poured into the tank. Salem Microbes, which is in India, has, again, products for poultry, or in this case in aquaculture, they're targeting shrimp. This case, they add them not as a feed additive for poultry, but in the drinking water. And for Vibrio, you can either put it in the pond or in a food supplement. And then finally, here in Asia, there's this company, CJ Bio, in Korea, that has products for poultry and swine, targeting either a mixture of bacteria or single bacteria, all as a feed additive. I'm not sure, actually, which countries they're selling in. They might even be selling in Malaysia. It would be worth seeing if we could contact and find out. So I want to go back to this. I don't think that you absolutely need, well, the data says that you don't need formal guidelines in order to move forward with phage therapy in different countries. Again, most countries have considered phages to be biologics that go through the standard biological regulatory process. And I think that there's enough guidance from other countries, and especially maybe with the stamp protocol applied here, that I'm not convinced that regulatory would be a major hurdle in Malaysia. I think a lot of the other things will take more time to put in place than any regulatory issues. And of course, let's go back to why we're doing this, and there may be more of an urgency for you to get this going in your country than even in our countries. I want to thank a number of people. So Drs. Hira and Siva at AIMST University, we started collaborating, I looked it up, in 2019, when we wrote a first grant together for a phage laboratory training workshop that we ran for people from throughout Southeast Asia, virtually in 2022 and then in person in 2023. And Rahil is one person from Malaysia who came and joined in that. And then in 2023, Siva and Hira invited me to come here to speak at a conference and introduce me to Prof. Sashila and Datin Shamani. Then more recently, when I came back to do the Fulbright a few months ago for 2 months, I was able to meet Drs. Aril and Dato Suresh, and then finally here I'm getting to meet Drs. Ami and Datuk Fariza. And I want to highlight a few. Dr. Cara Fiore at the US FDA has been hugely supportive of my Fulbright project and is very willing to meet with Dr. Zaril or other regulators around the world to talk through some of the issues they've had to think through. And then Dr. Jean-Paul Pierney at Queen Astrid Military Hospital was absolutely instrumental in getting that Belgian magistral phage in place. And Bob Blasdell serves on the European Pharmacopoeia board that's taken up the magistral phage for all of Europe. I really want to give a big shout out to somebody who hasn't been mentioned yet and who couldn't be here today, Dr. Carmen Coxon, who's at the regulatory agency in the UK. She is funding this symposium, really, through a grant from the Global AMR Innovation Fund, or GAMRIF. And so we're hugely thankful that she provided the funding to fly everybody in and house, and this would not be happening without her, so thank you to her as well.
Thank you, Yang Bruce Head, Dr. Toby Nagel. As rightfully mentioned, we are in the region which is heavily impacted and most vulnerable to AMR. So I think we have to catch up and keep up with research and development, which is actually one of the reasons why we hold symposiums like these, so that we can foster collaborations and capacity building moving forward. So ladies and gentlemen, our final speaker today is Yang Berusaha Dr. Zaril Harza Zakaria, who will be expanding on what Dr. Nagel just talked about. So Dr. Zaril serves as the head of the investigational product section at the Malaysian National Pharmaceutical Regulatory Agency, NPRA. With over 20 years of expertise, he plays a pivotal role in evaluating clinical trials and first-in-human or FIH studies. As a certified good clinical practice inspector, Dr. Zaril has conducted inspections of Malaysian and foreign bioequivalent centers, ethics committees, and clinical trial sites, including those conducting FIH trials. Today, Dr. Zaril will be sharing with us the regulatory essentials of clinical trials in Malaysia. Please welcome Yang Berusaha Dr. Zaril Harza Zakaria.
Afternoon. Thank you for your kind introduction. Right. So I think we go on with the last leg of the presentation. After this, we have some workshop, and then I will be involved as well. So let's go on. So my talk will be with regards to regulatory essentials of clinical trials in Malaysia. There will be a bit explanation of fundamentals of phage therapy and how we classify it. For me, the fundamentals are very important to classify whether which products of phage therapy or bacteriophage belongs to. Okay, these are the outline of my presentation. We talk about the introduction of clinical trials in Malaysia, what you need to apply, where does the MPRA's or regulatory authority governs or has the clinical trial oversight whenever you want to conduct clinical trials in Malaysia, and including phage trials, yeah, if you want to conduct phage trials in Malaysia. And then we talk about guidelines and legal requirements that you need to adhere to in order to conduct clinical trials in Malaysia, and requirement of clinical trial import license and clinical trial exemption application. What are they? Okay. Right. So this is the slide whereby you can know where we, or as a regulatory authorities, jurisdiction in. So if you can see here, okay, you can see at the pre-marketing and in the product life cycle, drug discovery, preclinical, clinical trials, regulatory overview, approval and launch, and post-marketing surveillance. All of the phases, yeah, especially where GXP plays a part, you know, good laboratory practices, GCP, good manufacturing practices, good distribution practices are all our jurisdiction, except for drug discovery. So if you are in the university doing R&D studies or research, you are safe from us. Okay. So under the CDCR, Cosmetic and Drug Regulation 1984, our regulation, Malaysia regulation, we have defined what is clinical trials. Okay? If let's say in the future you want to conduct clinical trials involving bacteriophage, this is very important, which which you will know what are the governance of clinical trials under our law. It states there an investigation or series of investigation on person conducted by under the direction or supervision of person, determining the safety, effectiveness and other effect of any product. Okay, so the keywords here is product. If let's say you're coming up with a product, your intention of doing your trials and research, the ultimate aims is to have a product, then it falls under our jurisdiction, and this is where regulatory authorities plays a part. And the terms clinical trials and clinical study are synonymous. Some say clinical studies, some say clinical trials. It all can be used interchangeably. So it all pertaining to your documentation. Right, so these are typical phases of clinical trials, applies to bacteriophages itself. If it goes to preclinical studies and then first in human studies, phase 1, phase 2, phase 3, drug regulatory review for the product, and also they have phase 4, which is post-approval studies. These are the guidelines and legal requirements that you need to adhere to whenever you want to conduct clinical trials in Malaysia. Again, I would like to stress the definition of product here. If you use what are the product, a drug in the dosage unit or otherwise for use wholly or mainly by being administered to human or animal for a medicinal purposes. Okay, and or a drug to be used as an ingredient for a preparation for a medicinal purposes. So the keywords here is medicinal purposes. Anything with regards to therapeutic claim, therapeutic indication, it falls under the definition of product. And some of you may wonder what is the definition of drug in our law. We have here in our SODA 1952, it states that includes any substance, product or article, whether you use in humans or animal, internally or externally, for a medicinal purposes. So meaning that any substances that are to be used for medicinal purposes constitute the definition of drugs. Okay, this is under our Sales of Drug Act 1952. So in terms of regulation of clinical trial products in Malaysia, whenever you have product in line, if you want to conduct clinical trials in Malaysia, there are two so-called exemptions by the form of clinical trial import license and clinical trial exemption. So I would like to highlight to all the participants or stakeholders here that for Malaysia, you do not need to conduct local clinical trials in our country whenever you want to register your product in Malaysia. Okay, if you feel that your products, safety, efficacy, quality data are sufficient enough to be registered, you can use the data that you have so far, yeah, to apply for registration approval in Malaysia. We do not mandate that every product need to have a local clinical trials in Malaysia. Okay. So back to the exemption, clinical trial import license. If let's say you want to import a product to be used for clinical trial purposes in Malaysia, you need to apply for clinical trial import license. And then clinical trial exemption, if you want to manufacture product locally for the purpose of clinical trials in Malaysia, you need to apply for this exemption, clinical trial exemption. Okay. So what are the products that require CTR and CTX? Product including placebo, these are very important. That's why my slides here are very technical. This is for the use of participant whenever they want to have a snapshot of the requirement, because sometimes for regulatory approval it is highly technical. Okay, so what are the products that requires CTR application? A product including placebo. If let's say you have a trial Bacteriophage, you want to compare with placebo. The placebo also need to have a CTIL or CTX. A product with a marketing authorization when used or assembled in a way different from the approved form. If let's say you want to compare it with a comparator, bacteriophage, you want to compare with standard therapy, antibiotic therapy, which is marketed in Malaysia, but you want to make it randomized clinical trial, want to make it double blind, randomized, modify the label. Okay, whenever the registered product has been modified, it is also considered as an unregistered product. Even though the active ingredient is registered with us, you need to apply. If you modify the label, you need to apply for the CTIL or CTX. And then a traditional product with a marketing authorization with an indication of traditional use when used for unapproved indication for therapeutic claim, herbal product with therapeutic claim. Also, you need to apply for CTI and CTX. Okay? And then, of course, an unregistered product, including placebo, manufactured locally for the purpose of clinical trials. Okay, classification of products. Now we come into fundamentals. Okay, if let's say your product, your phages, your bacteriophages, you have come upon the product itself. If let's say there's uncertainty in terms of your classification, I can see that, Toby explained to us that it can be, a majority of it are biological, but sometimes we need, as a regulatory authority, we also need to open up of quite a lot of claims. Okay, some say that even though it's bacteriophage, they have modified certain strain in terms of the product itself. If let's say you are uncertain about the product or bacteriophages, whether it's biological, CGTP, a cell and gene therapy product, or anti-MP, or otherwise, we have a section, we call it classification section, whereby applicant can verify the product classification with us and submit to us, and then we classify accordingly. So these are the two there are the form, the manual, the guideline, and the form. Okay. Right, these are the phage fundamentals. I always try to define it as best as I could. Phages and bacteriophages are naturally occurring viruses that play a crucial role in regulating bacterial population. So-called in the world of bacterial population, it is to make the condition a Zen, Zen condition. So in terms of its biological role, it attached to bacterial cells and inject genetic material. Diversity, it is highly diverse. It's found abundantly in soil, water, and human body. And application, as this symposium has rightly put, it is particularly used as an alternative to antibiotics. Yeah, there's a research been going on. So when it comes to classification, most of it are considered as biologic. What are the criteria? Derived directly from living organism with minimal manipulation. Example there, a bacteriophage isolated from natural sources. Example, sewage or soil that is simply purified for therapeutic use without any genetic modification. And most of it can be considered as drug. As usual, even though it's chemically synthesized, a drug, a phage product can be considered a drug if it is processed to achieve a highly defined and standardized composition that meets stringent quality specification. Okay, these are the consideration option, alternative. Okay, because when they come in for registration, they can come with quite a lot of claim. So the classification of the phages itself must be. We need to consider all sort of classification. And then it can become a CGTP or ATMP. CGTP is cell and gene therapy product, or NTMP is so-called, they call it in Europe. Involves advanced manipulation such as significant genetic engineering or ex vivo modification, which go beyond minimal processing. Okay, when you modify the strain, it involves major manipulation it can be considered as cell and gene therapy product or NTMP in the Europe. Okay. So back to the guidelines, if you have classified your product, so these are the guidelines that you need to follow. The first, the main guidelines is the Malaysian Guidelines for Application of CTR and CTX in Malaysia, 8th edition. And then a Malaysian Guideline for Phase 1 Unit Inspection and Accreditation Program. If, let's say, you want to conduct first in human trials in Malaysia, okay, and then followed by the 3 remaining guidelines for inspection, bioequivalent inspection, and ethics committee registration and inspection. The 3 remaining guidelines are pertaining to the whether or not you want to conduct bioequivalent study for other products. But the main 2 guidelines are Malaysian Guideline for CTR and CTX, the blue guideline on your right, and the purple guideline, Malaysian Guideline for Phase 1 Unit Inspection and Accreditation Program. Right, this a snapshot of the requirement of CTR and CTX. Who can apply? Any principal investigator can apply, or an authorized person from locally registered pharmaceutical company. Okay, we want a company from Malaysia to apply, or contract research organization with a permanent address in Malaysia. Okay, if let's say the CTR or CTX include a prescription or poison or drug, it must be submitted by type A license holder, which is a pharmacist in Malaysia. Okay, so apart from the form, you also need to provide the clinical trial protocol, a standard format, a pharmaceutical data. So some of you might wonder, where is bacteriophage comes along. Like Toby said, we have no specific guideline related to phages. If let's say your product falls under biologics, you can apply in terms of, fill up the forms pertaining to pharmaceutical product on the Appendix D6, which is biological investigational product in clinical trials. Okay, please pay particular attention in terms of these classifications. And then you need a GMP certificate from recognized regulatory authority, certificate of analysis, stability data, manufacturing data and formulation, and also product labeling. All of these need to be taken care of upfront whenever you want to apply to us. So another part which is very important, investigators brochure. I'm sure that if you are involved in clinical trials, you know what it is. Okay, this is a standard format for investigators brochure pertaining to the product itself, in this case bacteriophages, which include safety data and as well as efficacy data of that particular brochures, okay, of that particular product. Other requirements, you need that trial need to be registered with National Medical Research Register. You need to have an NMR ID, okay, and then ethics approval. Okay, you need both approval from relevant ethics committee and also to us, or Drug Control Authority, MPRA, must be obtained before initiation of any study-related activity at the trial site. Okay, this is what I explained earlier with regards to first-in-human application. If let's say your product, you need to conduct a first-in-human study in Malaysia. You have your preclinical studies conducted in other countries, but you want to conduct, it's not mandatory again. If let's say you want to conduct your first-in-human study in Malaysia, not only that the product need to apply these both exemptions, CTI, CTX, CTI or CTX, but also the site also must be accredited by us. The site, the hospital or the clinical site that you want to conduct the clinical trials, the first-in-human trial must be accredited by us. Okay. So, so far in Malaysia, there are 2 sites that have been accredited by us to conduct first-in-human study. One is at Hospital Sarawak General Hospital, and the other is Ampang Hospital. Okay, And then you need to provide to us the proof of insurance cover and declaration by the sponsor of application involving first-in-human clinical trials. Responsibility, it is responsible for the applicant, for the product to provide all the required information, as necessary. If you use a contract research organization, let's say if you have a conglomerate or company or sponsor from UK or outside of Malaysia, you want to use the service of contract research organization in Malaysia, a letter of authorization should be submitted to us. And again, I don't want to use this authoritative statement, but I need to, as a regulatory function, any person who knowingly supplies any false or misleading information commits an offense. So factors affecting approval. So the speed of approval will be dependent on how complete is the information submitted and how fast the sponsor provides to queries and adherence to established procedure, as well as ethical approval. So these are the timelines. So when you deal with regulatory authority, these are the timeline for us. If your product falls under biologics or first in human, it would take you about, for standard review, 45 working days. And for other product, simple molecule, it takes you about 30 working days. We also have a fast track review of clinical trial application. This is whereby you need to conduct a trial in the pandemic situation. For example, during COVID last time. This is whereby we can cut the timeline or the time of approval by half, by half. So these are the flow chart. So the one that I want to show you is that we are very transparent in our procedure. It follows the respective timeline that you need to that you expected from us whenever you submit to us until you get the license. Or the exemptions. Conditions, the approval is valid for 3 years. If you want to conduct a long-term study, you need to renew. It should be made within 3 months of expiry date. And then you need to adhere to drug accountability and disposal. You need to archive safely all the important information that you can read at the presentation. I hope that this slide will be shared by all the participants. This is very important and very technical. Okay, so thank you for your kind attention. Do you have any question? No question.
If anyone in the audience has any questions for Dr. Zariel, you're welcome to come forward.
Okay, see you at the workshop.
Thank you, Yang Berusaha, Dr. Zariel. I hope Dr. Zariel's talk just now has sparked encouragement and not hampered any of our interests to take up some clinical trials involving phage in the future. So as you can see in the agenda, we have the rest of the afternoon dedicated to our focus group discussions. But before we break up into our groups, I would again like to invite Yang Berusaha, Dr. Toby Nagel, to give a brief summary of today's session so far and to highlight the key takeaways from the remarkable talks that we've heard to carry into our collaborative discussions. Please welcome to the stage again, Yang Berusaha, Dr. Toby Nagel.
Thank you. So I have 5 minutes to summarize everything that we heard today. No problem, right? But I think maybe if we were to have an image, this might be the way to do it. That these are kind of the key components that you might consider including in a national framework for phage therapy. Starts and ends with pathogen surveillance. There needs to be an AMR surveillance system for your particular pathogen in your particular setting. Maybe it is Acinetobacter baumannii in CRC hospitals, something like that. Phage isolation, characterization, phage bank, and cocktail development, I think are all the things that would be discussed this afternoon by the Research and Biobank discussion group. Manufacturing, regulatory, and clinical application We're actually going to combine the regulatory and the clinical application groups this afternoon, since you need to talk to each other anyway to move things forward. But really, once you make it all the way through, then ideally it needs to go back to updated pathogen surveillance and updating phage cocktails. Have your strains evolved over the past year, just like we update a flu vaccine every year. Ideally, that might be how one would set that up. If Balint's data holds true and the bacteria is constant for 6 years, that would be great. Then you don't have to update your cocktail. But you need to do the surveillance to know that for sure. There. And so as we sort of think about going into discussion groups, what are some of the key questions that these groups might want to talk about? One is, I know that there is some AMR surveillance going at different sites in the country. How do you coordinate that all into a centralized way? How do you select which clinical indication and bacteria to target perhaps as a pilot? We've talked about Acinetobacter. That might be the right one, but I think let's discuss it. Let's make sure, is that really what you want to focus on? Phage isolation, characterization, and cocktail development, and the possibility of engaging a C-phages type program so that you utilize your student population as part of this process. And I could say that you might be able to fast track the cocktail development if it turns out that phages from somewhere else in the world, like from Hungary, could work here. Then maybe you could start with those as you also work to develop your own local phage cocktails. But I think long-term having your own centralized phage bank and database will be really valuable to the country. Many countries or institutions, leading institutions in different countries are now doing that around the world, starting to do that. We've talked about manufacturing capacity. I think ultimately COVID taught us that having manufacturing capacity in your own country would be really valuable. The vaccine manufacturing, that some resources have been, more resources have been put into that, I think, since COVID. Those could potentially be your infrastructure for manufacturing phages. We've talked about the regulatory framework. It sounds like that's going to work here. I don't think that's going to be a huge hurdle. I'm seeing a nodding of the head. So that, you know, I think there's questions and clarification that will need to be worked out as you go through the process, but I don't think that's going to be a major hindrance here. And then what might make sense, and this is for the groups to discuss, is do you want to go forward in parallel with compassionate use and clinical trial? Do you want to take STAMP as a way to do that as a first step, or something like the STAMP protocol? And then how would you go about updating the cocktails and incorporating that into the AMR surveillance in an ongoing way? And so I think the overall potential strategy might be to have this centralized national phage bank in the long term, or import cocktails from overseas in the short term. Those phages could be contributed by scientists from throughout the country, develop cocktails targeting the strains, the predominant strains found to be circulating. That probably is going to be more sustainable and efficient than going after personalized phage therapy in Malaysia, though that's for the groups to discuss. In-country manufacturing, and then possibly pursuing these in parallel. So I wonder if there's anything I've missed in those as sort of key points that the groups will want to be discussing this afternoon. Anybody stand out for anything? John?
Just one question, I guess, that is where the clinical community is at the moment.
That is a good point. That was left out. Public engagement in general, whether it's your clinicians or your consumers, be they the patients or the poultry farmers or whoever's going to use the phages. I do think public engagement is a hugely important piece of getting phages accepted. Okay, with that, I think we are going to break out into 3 groups.
Yes, okay, thank you, Yang Berusaha Dr. Nagel. So before we break for tea and adjourn to our discussion rooms, just a few quick important announcements. Firstly, regarding the group discussions. So the group list is provided in the welcome bag that you have received during registration. Please kindly take note of your assigned groups. However, please also do take note that we have merged Group 3 and Group 4 together for the discussions. So the new groups are now as follows. For Group 1, which would be discussing phage research and biobanking, the facilitators would be Associate Professor Dr. Jeremy Barr, Dr. Belin Kinses, Dr. Sivachandran Parimanan, and they will be discussing in Bilik Mesyuarat Pintar 3. So that's Bilik Mesyuarat Pintar 3 for Group 1. And for Group 2, we'll be discussing phage product manufacturing with their facilitators being Dr. Frank Smreka and Dr. Andrea Kwan Lehun in Bilik Mesyuarat 7. And Group 3, which have now merged with Group 4, will be discussing clinical application and regulatory framework. And the facilitators would be Professor Dr. Jonathan Iridel, Dr. Suresh Kumar, Dr. Toby Nagel, and Dr. Zaril Harza. And they will be discussing in Bilik Mesyuarat 8. So all of these discussion rooms are on level 4, but you may have your tea break first in the Day 1 banquet. So the objective of the group discussions is to address key issues related to the phage research as outlined by Dr. Toby Nagel just now, as well as therapy and application and regulation. And these points will be presented tomorrow. So the session is supposed to end at 4:45, or it could go on as long as you want to discuss, as you're free to discuss, after which you are free to adjourn. But I think we do have to lock up at 5 o'clock. So the second announcement is regarding the dinner tonight, which will be held at Marriott Courtyard Hotel. So once you've arrived at Marriott Courtyard, please make your way to the Five Wells restaurant on level 2 courtyard at the Marriott Hotel City Alam, and that will be at 6 p.m. And lastly, for tomorrow's group discussion, we will continue in the respective meeting rooms at 9 a.m. and reconvene for the rest of the session at 10 a.m. here in Auditorium 2. Thank you, enjoy your tea break, and have a fruitful discussion, hopefully.